I will allow.
Shankar, shall we start?
Yes, we are live now. We can start.
Okay.
So Prashant?
I'm really sorry to announce that Prashant is having some technical glitches. So on his behalf, a very good evening to all our distinguished guests, dignitaries, participants, and my dear lovely audience. It gives me immense pleasure to welcome you all to the second day of First International Forensic Science E-Conference 2021. Before we start the oral presentations, I would like to thank our scientific international experts, Dr. Emilio Nozzolisi, sir, Robert Scal, sir, Professor Michael Freeman, sir, Eugenio, sir, and Cloyd, sir, for their presence. First of all, I would like to thank you all for the trust and blessings that you bestowed on us. Thank you so much, respected sirs, for being there with me for our late night discussions and meetings regarding the event. Thank you so much to the international experts who kept their patience and were always there to support and guide me. I would further take this opportunity to thank our national experts who will be supporting our international panel for the evaluation of oral presentations. We are really excited to tell you that our scientific panel is very strong from India, USA, Netherlands, UK, LA, etc. I would also like to warn the oral presenters to be careful while presenting because the evaluation is going to be harder. Audience, we are looking forward to your comments, views on the presentations of our oral presenters and also your questions. You will be getting a big surprise filled with memories to take away. Let the energy flow through you as we start our event. Now, without wasting much of the time, I request Nandini to take over the platform.
A very good evening to all the international experts, Dr. Emilio Nozzolisi, sir, Robert Scal, sir, Professor Michael Freeman, sir, Dr. Eugenio, sir, and Cloyd, sir, and to all our national experts from the National Forensic Sciences University, Dr. Rakhi Agarwal, ma'am, Dr. Raj, sir, and Dr. Bhagav Patel, sir, for giving their valuable time for evaluating and analyzing our session of oral presentations. I, Nandini, with my co-host Ashika, on behalf of my whole team of the Forensic Corner, welcome you all to the second day of the first ever International Forensic Science E-Conference powered by the Forensic Corner and organized by the students of National Forensic Sciences University. Before we begin with our day 2, I would just like to once again tell about all our experts very briefly to our respected oral presenters and to all our audience who are watching us live on our YouTube channel. Our international panel of experts is a pillar of strength and support. Dr. Emilio Nozzolisi, sir, who is a forensic odontologist, researcher, and professor in legal medicine at the University of Turin, Italy. He is the president and founder of Civil Protection Association, Dental Team, DVI Europe, and co-founder and current president of the Association Forensic Odontology for Human Rights, AFOR. Moving on towards our next astonishing international expert, Mr. Robert Scal, sir, who is a consultant and a forensic fire investigator with the Gulf Coast Fire Investigation, Research and Education, LLC, based in Mandeville, Los Angeles. Our next experienced and proficient expert of the day, Mr. Cloyd Steger, sir. He is a former chief criminal investigator at the Washington State and Attorney General Homicide Investigation. He is also the Director of Investigation for the Midwest Cold Case Task Force, which assists in the investigation of cold case murders in the Midwest United States.
Moving on with the next distinguished personality to honor us with his presence is Professor Dr. Michael Freeman, sir, who is a tenured Associate Professor of Forensic Medicine and Epidemiology at Maastricht University Medical Center. He is a member of the Faculty of Forensic and Legal Medicine of the Royal College of Physicians, UK, and a Fellow of American College of Epidemiology. We are very fortunate to have amongst us a great forensic biologist, Dr. Eugenio Diorio, sir, who is the General Director of Bioforensics Research Center, a private Italian institute that performs research in the field of forensic biology in partnership with the University of Naples and different international forensic organizations and universities. From 2016, he is a consultant for the Italian Ministry of Justice about forensic DNA evidence. Let me further take this opportunity to once again introduce to you our national experts of the day. Annexing from Dr. G. Rajesh Babu sir, who is a well-experienced academician and researcher with more than 24 years of research and teaching experience with strong academic qualifications. He is currently the Associate Dean of School of Medico Legal Studies at National Forensic Sciences University, India. He has expertise in anthropology, advanced fingerprint sciences, terminal ballistics, and material sciences. The next intriguing expert that we are very fortunate to have amongst us is Dr. Bhargav Patel sir, who is a well-qualified and technically proficient academician with more than 10 years of research and teaching experience. His expertise include molecular diagnostics and microbiology, lab and field research work, data collection, analysis, and project management. Currently, he is an associate professor of senior scale in forensic biotechnology at National Forensic Sciences University, India. Moreover, we are blessed to have amongst us a renowned personality in the field of toxicology, Dr. Rakhi Agarwal ma'am, who holds more than 17 years of professional experience, including about 12 years of academic experience in the field of toxicology. She is currently serving as an associate professor at National Forensic Sciences University since university inception, that is July 2009.
As rightly said, don't be afraid to try something new. Mistakes are an important part of learning process. With all our best wishes to our dear presenters, I hope that by now you all must be very much excited and ready to present your work in front of a scientific panel. Before that, I would also like to tell the rules of oral presentation. I request the oral presenters to wind your presentations in 8 minutes, and we will give you a reminder after 5 minutes. I request all the presenters to follow the rules properly and abide by the time limits. Otherwise, your presentations will not be considered apt for evaluation. And in order to maintain the decorum of the event, any unnecessary disturbances will not be entertained. So I request the oral presenters to respect the event norms and the fellow colleagues and our scientific panel. Further, I would also request our respected audience to keep on commenting for their questions, feedback, as you all know that the best comments will be getting big surprises. And we have kept 2-minute session for the question answers round from our international experts. Without any further ado, I would like to start the event by calling our first presenter, Ms. Jasra Rahman, who is a student of LNJ National Institute of Criminology and Forensic Sciences, India. She will be presenting on the topic, Effects of Medium of Primary Education on the Learning and Usage of Latin Script. Over to you, Jasra.
Jasra just informed that she's having a technical issue, so you can proceed with the second participant.
Okay. Yeah, sure. So our next presenter is Mr. Shubham Awadh, who is a student from Yashwantrao Chavan Institute of Science, Satara, India. He will be presenting on the topic Sexual Dimorphism from the Visual Examination of Tongue in Satara District. Over to you, Shubham.
Yes, should I present my screen or
Yes.
you will be present?
No, you can do.
Yes. Is my screen visible?
Yes, it is. All the best.
Yes, so good evening, everyone. I'm Shubham Awadh, of the student of third year BSc Forensic Science from Yashwantrao Chavan Institute of Science, Autonomous, Satara, Maharashtra. So the topic, research topic of my research topic is the sexual dimorphism from visual examination of tongue in Satara district. So the aim of my study to study sexual dimorphism from visual examination of tongue in Satara district. Objectives are to examine morphological features such as shape, length and width, presence or absence of fissures on the tongue, and to study sexual dimorphism on the basis of visual examination of tongue in Satara district. The introduction to my research. We all know identification of individual plays a very important role in crime investigation, mass disaster, and any disease cases. And there are many traditional biometrics like fingerprint, face, palm print, and voice also. And but they have inherent limitation as they are can be easily forged. So the tongue is a vital organ which is well enclosed within the oral cavity, and it is unique to each person in its surface characteristics, mainly shape, its texture and size. And the uniqueness of tongue may aid in individual identification and can also be serve as a new biometric tool. So the methodology of my research was I took 200 study participants from Satara district, and where and in they are healthy subjects comprising of 100 males and 100 females between 11 to 30 years old. And after I took their visual photographs of tongue after taking their written consent, and then the digital photographs I took by using a 48 megapixel quad cam, and the data I collected were subjected to statistical analysis using chi-square test. And in this research study, I consider three morphological characteristics: the shape of tongue, border of tongue, and fissure of tongue. In result, in table 1, there are two groups. In group 1, there are 100 males, and in group 2, there are 100 females. The second table, shape of tongue. So in males, by analyzing the visual photographs of tongue, the bifid tongue in male are 21, the U-shaped tongues are 51, and V-shaped tongues are 28. And in females, the bifid tongues are 26, U-shaped tongues are 29, and V-shaped tongues are V-shaped 45. And after the analysis by using chi-square test, I analyzed the data. In table 3, the borders of tongue. The partially scalloped borders in males are 39, scalloped borders are 26, and smooth tongues are 35. In females, the partially scalloped tongues are 44, the scalloped tongues are 19, and the smooth tongues are 37. And also in table number 4, the fissures of tongue. In males, there were in 44 tongues, there are no any fissure present, and the tongue with multiple fissures are 15, and the tongue with single fissure are 41. And in female, the 61 tongues are don't have any fissure, and 16 tongues having multiple fissures, and 23 tongues having single fissure. As we see in photographs, I took visual images like this. In first image, as we can see that bifid shape of tongue. In second image, the U-shaped tongue and V-shaped tongue. And in between the middle image, the partially scalloped tongue, and smooth tongue. And in last, in first tongue, there is no any fissure present. In the middle, there are many fissures, and in last, the single fissure is present. So the discussion in the shape of tongue, the association between the shape of tongue and the gender was found to be yes.
Mr. Shubham, this is a reminder. Please conclude. Only 3 minutes are left.
Yes, ma'am. So the shape of tongue, the association between shape of tongue and gender was found to be statistically significant as I used the chi-square test. And the U shape, I found that U shape was predominantly common in males as compared to females. And the V shaped tongue was more common in females as compared to males. The reason could be a smaller mandible size as compared to males. Second, borders of tongue. The association between borders and gender was not statistically significant. The scallop border was observed more in males as compared to females, whereas smooth borders were observed more predominantly in females as compared to males. And the scallop tongue was observed more in males compared to females. The reason could be more parafunctional habits in males. And the last, fissures of tongue. The association between fissure and gender was found to be statistically significant. The multiple fissures observed more in females as compared to males, whereas single fissure observed more predominantly in males as compared to females. And the fissures are simply a variation of normal tongue which appear on the dorsal surface of tongue. And the conclude the research of my research topic. The present study represent the preliminary method for analyzing the morphological characteristic of tongue by visual examination between male and female in Satara district. The surface characteristics of tongue show significant variation in male and female and could be served as aid in sexual dimorphism. The lingual photographic image can enhance personal, personal identification along with other techniques in forensic science. And also there are many biometrics have been used and developed, but there is not much work done on tongue for personal identification. So thank you. I would like to take your questions.
Thank you so much, Shubham, Mr. Shubham. Now I would like to ask our scientific panel if they have any questions for him.
I have a quick question. I may.
Yes, sir.
Yes, sir.
Interesting presentation. Have you thought about a multivariate model where you could use more than a single character to model the probability of a particular gender?
Yes, sir. I took three characteristics: the shape of tongue, border, and fissure of tongue.
Where you put the two together?
No, I took 200 samples, and I analyzed particularly I took, for example, I took one single tongue, and then I analyzed the shape of this tongue, which borders are present and which fissures are present between particular tongue. So I analyzed like this for 200 tongues.
Thank you.
Yes, sir. Thank you.
Any more
I have a
questions?
question.
Yes, sir. Yes, sir.
From what you know, it is existing a reference sample for tongue evaluation phenotypical characteristic, or it is still not, and maybe it must be developed.
Yes, sir. Sir, can you please repeat your question?
Yes. From what you know, from the research that you did, also in scientific literature, is existing a reference sample for human tongue for identification purposes, or still not? And maybe the scientific community must work for developing these if it can be useful for identification and forensic applications?
Yes, sir. We can develop a particular database for tongue, and by using we can use for them for personal identification, for any mass disaster, and in also any disease case also. So it is a good idea to make a database for tongues. As we know, we have database for DNA, fingerprints. So it will be helpful to make the database on tongue prints.
Definitely agree. Congrats for you, the organization of your research.
Yes,
Thanks.
sir. Thank you. Thank you so much.
Do we have any more questions from our scientific panel for Mr. Shubham?
Okay, so let's move on to the next presenter. Our
Yes,
next,
ma'am. Thank you.
our next presenter is Mr. Nadim Khalil, who is a student from National Forensic Sciences University, India. He will be presenting on the topic dermatographics, a ratio study of North Indian Muslims.
Good evening, Nandini. Can you please share my presentation? I'm not able to share my presentation. I have some connectivity issues.
Shankar, can you please share?
Sure, sure.
Thank you.
I hope it's visible now.
Yeah, it's fine. So I'm starting. Good evening, panel, scientific panel, and good evening to my colleagues and presenters of this International Forensic Science Conference. I'm Nadim Khalil, and my research topic is dermatoglyphicus ratio study on North Indian Muslims. And I'm pursuing my master's in Homeland Security and Anti-Terrorism from National Forensic Science University. And I have pursued my bachelor's in Forensic Science. Next, please. Introduction of this dermatoglyphicus, my research topic. The whole research is revolving under the fingerprints and their issues. The surface, as most of the people know, the surface of human foot and face is covered and decorated by the layer of skin that's completely different from the other parts of the skin, and it's screwed with a minute ridges, and we call it fingerprints. There are no oil glands and hair glands present on these parts of the skin, as they are controlled by the polygenic traits. That means they are under self-control of additive genes with equivalent effects. The importance of the fingerprints is that they are a unique and reliable evidence in forensic science, which has been used in criminal justice system and accepted overall globally. Due to its unique name, which has been identified to the people, identity, this very unique identity to the people. The main two properties and the basic, we call the pillars of these fingerprints, are the fingerprints. Every fingerprint is unique to an individual, and fingerprints don't change all through the life unless and until something happened or the dermal layer is corrugated by someone, by accidentally or by intentionally. A quotelet statement that every nature made figures have infinite variations and no patterns are ever similar. This is the almost another fundamental about fingerprints. Next, please. Research question, and the basis of my research is that the present studies was carried about to determine the frequency of various fingerprints and statistically correlate with the correlate with its gender differences and with other ethnic groups also. Fingerprint samples were collected from the North Indian states, Delhi, Uttar Pradesh, and Jammu and Kashmir. The motive of this research was to determine the fingerprint ratio called as dermatoglyphics ratio in North Indian Muslims, then examine, analyze them, and then compare with other ethnic groups. The other purpose of research is to examine the peculiarities, how North Indian Muslims are different from how their fingerprint ratios and how their fingerprint patterns are different from others, which in future will help in criminal activities and to solve the criminal cases. Next, please. Literature review of this research is that I don't know other research have been done like that in North Indian. This is our first attempt, but in Central Indian it was done by the first attempt was done by Ashish and A.T. Kapoor. In this research, they have researched on Central Indian Muslim populations about 480 samples, and what the results got, they got 50.25% blue patterns, Wolves 28%, and composite 17.33%, and arch 4.42%. And Nitin at if we can correlate this research, Nitin's research with mine, and with others, it's somehow relevant because they got 500 samples, but they got with mixed communities in South India. They put their research on gender base. They differentiate between the genders and find that ulnar loops are more than in females than males. That 55.2% in females and 32% in males, and more whorls in males than females, 26.84 in females and 30.64 in females. Next, please. Methods of data collection, how we collected this data. I can tell you that the 150 Muslim subjects, 100 male and 50 females, over the North Indian, Delhi, Uttar Pradesh, and Jammu and Kashmir states. The age ranged from 6 to 58 years. The method of fingerprinting was not easy. We had to take the samples with the process. First, we have to wash their hands, make them dry, and after 5 to 6 minutes, we rolled their fingers from left to right and take their impressions on fingerprint 10-digit fingerprint slips. The process was somehow simple and sophisticated. We neglect the extra smudging of ink on the silk and marking silks with their age, gender, and place of residence. Next, please. Data presentation and findings. Almost a total of 100 and 500 fingerprints were studied and patterns identified overall. What the research, what we got, that the pattern loop is 50. Okay, 57.23% of our worlds and 43% arcs and 2.2% composites. Subclassifying that ulnar loop was 46%, and basically what we find that there was the main part of this, the main part of the research, that there are no lateral loop pattern found in the genders, and accidentals. Next, please. Accidentals were 0.13. This is the whole pie chart of my data, that ulnar loop 47%, accidental 0.13, worlds 35, concentric 8, radial 6, and tented arch 1%. That there is no lateral loop found in the North Indian Muslim populations. Next, please. The conclusion of this study is a detailed frequencies, whereas we were told, as I included, that Muslim populations, we take it and we find peculiarities, that we don't find any lateral pocket loop and accidental loop in 0.3%. A SATS analysis of t-test were applied to obtain results. Gender differences were found to be statistically insignificant and p less than 0.05. Thanks. And any questions?
I would request our scientific panel to if have any questions they can ask Nadeem. Respected sirs and ma'am, do we have any questions for Nadeem?
I think there are no questions, so we can move to the next.
Fine.
So moving on to our next presenter. Our next presenter is Dr. Kushali Joshi, who is a representative from IEIF India. She will be presenting on the topic forensic applications of EDXRF to identify gender from saliva and urine.
Hello, good evening, everyone, and good evening, the scientific panel and everyone, all the participants. Is my screen visible?
Not yet, ma'am. Yes, ma'am, it's visible now. Yes, it is.
Hello, everyone. I'm Dr. Kushali Joshi, and my topic is forensic application of application of EDXRF to identify the gender from saliva and urine. So basically, EDXRF is electrodispersive X-ray fluorescence. So as we all know that traces of body fluids recovered from the crime scenes are among the most important type of evidences to the forensic investigation, and it contains very valuable information like origin, the nature, or gender, and many things. The important aspect of them are detecting the gender identification, and driven by the importance of the forensic application, the body fluid identification methods are extensively developed. So there are certain techniques that are available, like DNA techniques, but the problem is a DNA profile obtained from crime scene cannot be associated to its source unless the person's DNA is already in the database. as a reference. So when there is no such information is available, it would be helpful if the evidence can provide information to the donor. So such information would narrow down the search for the suspect or the victim, and the information will be immensely helpful if it is available at primary stage or in the investigation, like immediately or in the crime scene. So saliva and urine are two body fluids which are maybe available on such cases like sexual assault or homicide or child abuse, kidnapping, bite mark, jogging, hanging, assaults, so many cases. So basically, the level of metallic components depends on the various factors like food or habitat, hormones, protein digestion, and many other things. And when we examine it with the use of EDXRF, so this is the principle of EDXRF. So when XRF is based on when excited by the external energy source, it will emit the X-ray photons of the characteristic energy of wavelength. So by counting the number of photons of each energy emitted from a sample, the elements present may be identified and then quantified. So that's why here I have taken 75 males and 75 females, total 150 subjects, the age group of between 18 to 30 years. So here this cotton will be put in the sublingual area of the subject, and then it is allowed to soak the saliva for 1 to 2 minutes. Then it is taken out and put in the syringe, and then it will be squeezed out in a plain tube. And the urine samples are collected freshly in the uricol. So these collected samples are loaded on the EDXRF to find out the various metallic components values with the use of Mylar sheets. And approximately 0.5 to 1 ml of sample is sufficient to get the results. This is the EDXRF, and like this we can get the results. And this is the portable EDXRF. So this method we can use on the crime scene or on the spot itself to get the results. So when we done the study, the main components which we found from the EDXRF spectra of saliva is potassium, calcium, silicon, and copper. And from the urine, we found potassium, silica, calcium, and sulfur. So these are the differences in between the male and female salivary metallic components. Here we can see the differences, and these are the differences in between male and female urine metallic components. Here we can see the differences. And in our study, we include 75 males and 75 female saliva and urine samples, and when we use the ROC curve on them, so in which almost 89% of samples has detected as a true positive, which means the correctly identified samples. So this study can be used as a presumptive test for the identification of the gender. And importantly, the results are available immediately or on the spot. And this technique is completely a non-destructive technique and without subjecting biological fluids for any sample preparation. So that directly we can take the sample from the crime scene and subject it for the EDXRF examination. So this is the first time that the biological samples are subjected to evaluate by its physical properties like metallic components present in the saliva and urine by the EDXRF technique. So this technique can also consider as a time and very cost-effective and reliable instrument technique. And in many criminal cases like crime against women such as sexual assault, child abuse, physical assault, or attempted homicide cases, bite marks, or so many cases. Yeah. Okay, so evidence is collected from such indicates that of saliva and urine. So this study has been done on equal number of population of male and female, that is also age group in between 21 to 30. So in addition, people suffering from various diseases or any habits like smoking or alcohol or drugs or those people are excluded from the study. So one can appreciate that 150 samples could not represent the adequate of the diversity of the population. Therefore, the study should be considered as a preliminary or proof of concept study, and it will be necessary for a larger and much diverse population for the further expansion of the study, increase overall robustness of this method. Thank you so much.
Thank you so much, ma'am. I would also like to ask our audience if they have any questions, they can comment. We'll be taking their questions as well. Meanwhile, I would request our scientific panel to ask questions, if any.
Khushali?
Yes, ma'am.
Yeah. Khushali, nice presentation and good work also. My concern
you.
is related to the old saliva stain. If some saliva traces or the stain has been recovered from the crime scene. So yeah, this method will be applicable there. Did you try that?
Yes, ma'am. If only stains are available, we can take a normal cotton swab and take distilled water, and we can take it by the swab method, and then you can squeeze it with the same method, what we have done in the subdivisional method.
You mentioned this already in your presentation for the old saliva.
No, ma'am. Not for the old saliva.
Okay. So
I mentioned only for the fresh saliva.
Okay, so what is the statistic evaluation? Did you perform this experiment with the old saliva or aged saliva? Okay, but
No,
you are just giving an idea that with this method it can be done. Okay,
yes,
fine. So what is the statistic evaluation, like the samples that you have examined, the fresh samples that you have examined? So what was the, I can say, the standard deviation or standard error with your results? What was that?
Ma'am, I have done with the standard deviation, and then I applied with the ROC curve. So in the R software ROC curve, we find out that 89, we got the true positive rate, that is correctly identified as a female or a male. Then 11% we got the false positive result. That 11% they are the method is identifying false positive rate.
Okay, good. Nice presentation. And one suggestion is there. Whenever you use one technique for the method development or for anything that is going to be new for the scientific fraternity, then you have to validate your method or the technique with another already known method or renowned. Like with EDXRF, you can compare the data with the atomic absorption, or more prominent or more promising instrument is the ICP. So there your data will be validated. So for future, since you are in the preliminary stage of your study, but for future, you can go ahead, you can think for that also. Thank you.
Thank you. Thank you so much.
Any more questions from the scientific panel? Okay, so ma'am, we have a question from you from YouTube comment section. The question is from Sneha Narayan. She says, would this EDXRT technique gives the same results in pregnant women too? Were they taken into consideration?
I have not taken any pregnant women, but I think if we go for the pregnant women, their metallic components may be different from the normal women, because during the pregnancy, the internal physiology will be imbalanced. So yeah, maybe I'm thinking that that it will metallic components level will be different compared to normal female.
Okay, thank you so much, ma'am. I hope this must have addressed the doubts of Sneha. Now moving on towards our next presenter, who is Dr. Pankti Patel, who is a student from National Forensic Sciences University, India. And she will be presenting on the topic, the emerging era of digitalization in forensic odontology.
Okay, am I audible?
Yeah, you are.
Okay, I'll be sharing my screen. Is it visible?
Yes, it is visible.
All right. Good evening, good afternoon, and good morning to the respective scientific panel and my fellow colleagues. I'm Dr. Pankti, and I'm a student of MSc Forensic Odontology here at NFSU. And today I'll be just giving a review about the digitalization in forensic odontology. So now what is basically digitalization? Digitalization refers to enabling or improving any process by leveraging the digital technologies and the digitized data. So to simplify it, it is a process that takes a human-driven event to a software-driven approach. And this increases the productivity and efficiency, and it reduces the human error. Now, digitalization in forensics has replaced the traditional investigation methods in terms of acquisition, the analysis, as well as the reporting. And similarly, the situation in forensic odontology has also shifted from utilizing the conventional methods to now, accepting the more digitalized methods. I'll be just highlighting the change, the shift in the digitalization which has been taken place in forensic odontology. So now starting with the disaster victim identification. Previously, there were odontograms and handwritten charts which were utilized, and now we have shifted to the digital dental charts, which are easy to report, and they can be uploaded and they can be shared easily. Human identification from the smile photographs have also been considered, in which you can use the superimposition of the incisal contours of the front teeth, and this data can be uploaded and it can be also shared. Similarly, a database which has been created for the comparison of the antemortem and the postmortem analysis, and the latest being the plasma data, which has been approved by the Interpol as well. New studies have also been found regarding the artificial intelligent neural networks, which has been utilized using the ANN and the CNN based models. And these studies are yet to be validated. But yes, there are scientific researchers which have supported the studies which can be used for age, sex determination, as well as bite mark analysis. Now coming to facial reconstruction. Facial reconstruction using computerized methods is less time consuming. It is more precise and it eliminates the human errors. And there are multiple softwares which can be used. Some of them are open, some of them paid. The steps which are generally involved in the conventional methods can be easily replicated in the computer aided forensic facial reconstruction as well. And this also helps in working with the multidisciplinary approach in case you want to share your data. Now, personal identification. Personal identification using your lip prints, your palatal rugae, and the bite marks. Now, chyloscopy and rugoscopy have been studied previously where the superimposition techniques have been used using the Adobe Photoshop. And for the bite marks, now bite marks have been a controversial topic in forensic odontology, which has been questioned by a lot of scientific researchers, but yet there are supporting studies. And two of the best studies which I found were is the GeoMem system, which is actually considered as the gold standard for the bite mark analysis. And one study is this 3D photogrammetric study, which has been used in combination with the laser scanner. This study is yet to be validated. Digital photography has been used to document the cases, especially in case of abuse marks or your bite marks. And we also use different type of lights, for example, UV, infrared, and your alternate light imaging. The same lights can be also used to study the different restorations. So if you want to, in case you need to differentiate between ceramic and, the composites, the same lights can be used. The digital photographic system basically works on your CCD and CMOS sensors, and they also have inbuilt software which will convert the digital file, and it can be directly transformed into the computer. Now the vitropsy is being used now more frequently because of the COVID pandemic situation, and this is a great tool for your DVI investigations, because it helps in the teleconsultation as well. So basically, you first surface scan the body and you place some certain markers which can be color coordinated into the mesh. And your CT and MRI scans, the CT and MRI scans are taken simultaneously. And the entire data is...
I request you to wind up quickly. 3 minutes left.
Okay, sure. Yeah, and the last is the use of 3D scanning and 3D printing, which is also being considered nowadays in the forensic odontology, which can help you with the craniofacial reconstructions, your superimpositions, your age and sex determination. It can also help you to illustrate bone pattern injury. So I would just like to conclude that the emerging era of digitalization has opened a lot of scope for new research and creativity in the field of forensic odontology. And there are things which need to be validated. And in the upcoming years, it will definitely be a part of the odontology research community. So these are my references. Thank you very much.
Thank you so much. Now I would like to ask our scientific panel if they have any questions they can ask. I would also request our respected audience to please keep commenting. I think we have a question for you in the chat box. Okay. Small correction. Yeah, there's just a small correction from Michael sir that it's word topsy, not word topsy. What you have written? You have written word topsy. It's word topsy. He says.
Okay, yeah.
Hi, Pankti.
Yes, sir.
I have a question actually. I want to ask whether we are having in India, is there any dental database that is already present, or are we working on development of any dental database? And apart from that, if our dental database is being prepared or is to be prepared, what are the dental features that can be added which can aid in forensic investigation?
Yes, sir. So now currently, so there is not an integrated dental database which is present. There are private dentists which keep a digitalized form of data, but we don't have an integrated data which can be shared or which can be networked or which can be linked with the Interpol. So in case if we are preparing a dental database, the same odontogram, which includes the number of teeth, the kind of restorations which are being done, the radiographs which are there, the same can be replicated digitally, and it can be saved. So in case if you need the data for any identification, or you need to compare it, this can be shared. The basic purpose is you can share it.
Okay. Well, you have raised a very good topic, to be honest, and it's going to be of a great importance, like in future, if you talk about criminal investigation. So all the best.
Thank you, sir.
Okay, so we have one more question from the YouTube audience. What is the legal stand on 3D printing?
Legal stand on 3D printing, as far as in India, there is no legal stand. So if you want to present evidence which has been 3D printed, they won't accept it in the court. But yes, there are researches which support the studies. There are scientific researches. Nothing has been actually considered legally. But yes, there is a lot of scope, and there are a lot of scientists who are working on this. Like if you want to digitally present evidence in the court, so it actually depends upon the legal system of different countries. As far as I'm talking about India, it is not acceptable yet. It might be in future, but not yet.
Okay, thank you so much, Dr. Pankti. Now I would like to move on with the next presenter, who is Mr. Kushal Singh, who is Assistant Professor from SJT University, Haryana, India. He will be presenting on the topic Sweat Testing for Nicotine and its Metabolite Cotinine by HPTLC-MS, an examination to assess the possibilities of sweat as an alternative matrix in forensic toxicology.
Good evening, everyone. Am I audible?
Yes, sir, you are audible.
Okay. Is my screen visible?
Yes, yes.
Okay. Good evening, everyone. I'm Kushal Singh, working as assistant professor in SGT University. So today I'm going to present my part of research, that is sweat test for nicotine and its metabolite cotinine by HPTLC-MS, which is an examination to assess the possibilities of sweat as an alternative matrix in forensic toxicology. So as we know, in forensic toxicology, basically viscera is our traditional metrics for drug identification. So these days, with the modern instrumental analysis, sweat is a modern biological metrics for detection of drug of abuse. And the basic function of sweat is to thermoregulation, basically. In comparison to traditional metrics like urine and blood, sweat have upper hand in terms of non-invasive sample collection and less hindrance from metrics and other impurities like protein. Along with that, sweat provide both narrow as well as long window for the consumption of drug as compared to saliva and hair examination. So for hair examination, you can detect the drug from weeks to years until unless you don't cut the hair. Whereas in saliva, you can detect the drug and its metabolite within hours to weeks. Whereas sweat have both, you can go for a long as well as short window of detection. Due to the acidic pH of sweat as compared to the blood, basic drug easily accumulate in sweat as per the pH partition theory. And as we know, 90% of drug we encounter in forensic are basic in nature. So that's a very important thing. Here you can see the structure of nicotine and its metabolite cotinine, which is converted by the enzyme, which is the cytochrome P2A6. For this study, nicotine is selected due to the ease of availability of sweat sample and to ascertain that the nicotine consumption, one major metabolite marker, that is cotinine, is also included in this study. The purpose of the present study is basically a method development for nicotine and cotinine analysis from sweat sample. Detect them by various techniques which were never tried before, like HPTLC, MS, GLC, and TLC. So in this topic, I will discuss about the HPTLC MS only and apply these developed method on sweat sample for simultaneous analysis of nicotine and cotinine in single sample. So as you can see, the difference between nicotine and cotinine structure is just the difference of one keto group. So what I have done, first I've collected this sample from normal swabbing with the filter paper, which is dipped in ethanol and distilled water in 1:1. After that, I applied these extraction method. After that, I went for TLC analysis, and after TLC, I did the UV visible spectrophotometer. So for HPTLC analysis, first I need to develop a solvent system and absorbance. So for that, I did TLC and UV visible. And then that sample was analyzed in HPTLC, and those positive samples, those who give positive result in HPTLC, I analyzed with HPTLC MS. So I totally collected 50 samples, 2 sample of non-smoker, 11 sample of smoker, which include active and passive smokers. In active smokers, I took BD smoker, which is a traditional smoking stick of India, and 5 sample of cigarette smokers. Along with these, I took 2 control sample. One is positive control, which contain the standard cotinine and nicotine, and 1 negative sample to study the interference from mattress. As far as for the extraction, I applied 3 extraction method, that is liquid-liquid extraction, direct solvent extraction, and SPE method. With LLE and SPE method, I give 2 conditions. So total, I have 5 extraction method, that is LLE1, LLE2, direct solvent extraction, SPE1 and SPE2. So total 5 extraction method were applied to 15 sweat samples. So therefore, I have 75 samples for the analysis. Result and discussion. So this is the solvent system which I developed for the analysis of nicotine and cotinine in a single sample. I use drag and drop spray reagent and isoplatinate, and these live sample were analyzed with isoplatinate spray reagent only. These are the UV visible spectrophotometer results of nicotine and cotinine. Nicotine shows the absorbance at 261 nanometer, whereas cotinine showed the absorbance at 262 nanometer. After that, I use these data for the HPTLC analysis. Here you can see the HPTLC plate at 254 nanometer fluorescence.
Sir, I request you to wind up quickly. 3 minutes only left. Okay,
ma'am, sure. So here you can see the RF value of cotinine, which maximum RF is 0.2, whereas for nicotine, it's 0.8. And those positive sample I analyzed in mass. So nicotine shows the M by Z peak at 163 and 193, whereas cotinine shows the M by Z peak at 106. So conclusion of my study is that with TLC analysis, as I told you, cyclohexane toluene diethylamine with 75:1, 15:10 ratio solvent was developed by me. There is no such solvent system for nicotine and cotinine. And all the extracted sample were analyzed in this developed solvent system and isoplatinate spray reagent. SPE1 shows the better result. And for developed solvent system, this is used for the HPTLC analysis. Again, here SPE1 is the shows the more promising result as compared to other extraction method. And the MS result was for nicotine was 163 and 195 at RF 0.6, whereas for cotinine it was 106 at RF 0.2. So these were the references. Thank you so much.
Thank you so much, sir. Now I would like to ask our scientific panel if they have any questions. So we have a question in the chat box for you, sir.
Okay, ma'am.
Do you have information on the half-life of cotinine in sweat? How do you envision this technique being used in a forensic setting?
I have no idea about the half-life basically for cotinine. Basically, my aim was to study the sweat as a matrix, as an alternative matrices. I took nicotine and cotinine because of the easy availability of a collection of sample. So.
Dr. Kushal? Yes, Dr. Kushal?
Yes, ma'am.
A nice presentation, nice work also, but I have few concerns.
Sure.
Did you go through for the publication process? You are looking for the publication of this work?
Yes, ma'am.
Okay, so my suggestion is that I'm not sure, but that I felt your RF is very close to the sample origin point, and it is just 0.17, if I am right.
Yes, ma'am.
Yeah, so this is very close. Try to change your solvent system. The solvent system. Can you go to your back to your slide?
Ma'am, is it visible?
For me, it's not.
Okay,
just tell me your solvent system.
Ma'am, my solvent system was cyclohexane toluene diethylamine. Actually, ma'am, I used 10 different solvent system, and this was the best. Otherwise, all the solvent system shows the difference around 0.02 or 5. That's it.
No, whenever you go with the TLC, you
Yes,
have to have the combination of the polar and the non-polar solvents.
Yes,
okay, so the same system, if you will change the combination, like 75 is to 15 is to 10, something is your the amount that you have taken, percentage that you have taken.
Yes,
so just play with that, and you will get the separation in a good manner, because your bands are so close that I can understand from your presentation.
Okay,
so while you will go for the publications, this will be one of the major hundreds for your publication.
Sure,
okay,
sure, ma'am.
so just go to the literature and try to find out the best solvent for the alkaloids, and then try. So this RF should be at least above to 2 or 3.
Okay, ma'am. Okay.
0.2, it should be above to 0.2 or 0.3. Okay,
sure,
and there must be a difference.
Okay.
Similarly, your UV data is not showing a good resolution. If there will be no good resolution, then if the as such nicotine is present and the metabolite is present, so the peak will be merged.
Ma'am, actually, I did a UV analysis for live samples also, but they didn't show any result. It just for the HPTLCMS for the density, so that I can see the absorbance. So I use that data in HPTLC.
Actually,
I see the absorbance is 261 nanometer.
Okay, fine. This is not my question. This is for suggestion your future.
Okay, ma'am. Yes, ma'am.
Okay,
sure,
fine. So all the best.
Thank you so
Thank
much.
you. Over to you, Ashika.
Ma'am, sir, we have one more question for you. If
sure,
suppose the person has taken 2 or more drugs, wouldn't it result in a difference in the results obtained? And so how reliable is the method when we have no idea about the person? So this question comes from a YouTube from Sneha.
Ma'am, if you have no idea, you need to have like, by mass you can identify basically by fragmentation. So first you need to develop the TLC system. And if you have no idea, then, you are aiming in the, a black box. So you need to have a bit idea about it, the consumption of drug.
Okay. Thank you so much, sir.
Thank you, ma'am.
Our next presenter is Ms. Sakshi Manhas, who is a research scholar from SGT University, Haryana, India. She will be presenting on the topic molecularly imprinted polymers, a boon for detecting trace components in forensic science.
Hello, good morning, good afternoon, and good evening to the respective individuals. There is one correction. I am a research scholar. My name is Saffron, and I am doing my research in lovely professional university.
Okay, ma'am, my apologies.
Yeah. Thank you so much. Am I audible clearly? My presentation is visible?
Yes, ma'am, it is visible.
Thank you so much. So, just a minute, I'm displaying the presentation. Yeah. So, hello everyone. My name is Sakshi Manhas. I'm a research scholar in Lovely Professional University. The topic of my presentation is molecularly imprinted polymers, a boon for detecting trace components in forensic science. So here I intend to outline the significance of MIPS, that is molecularly imprinted polymers, in detecting trace elements from forensic science to meet the current need of criminal investigation with efficiency. So coming to the field that is forensic science, as we all know, it's the application of extensive ambit of sciences and scientific fields to criminal investigations and legal proceedings. Forensic analysis mainly consists of detection and comparing of the question sample with the known sample. In this field, the most challenging aspect is to detect and identify trace elements from the complex mediums. These elements can be drug, DNA, explosives, etc. So forensic science is a growing field which needs effective analytical techniques, and MIPS can prove to be a great significance towards modern day research. Also, the major limitations of using the conventional techniques are specificity, selectivity, short shelf life, getting false positive results, and the instruments are not cost effective. Molecular imprinting involves designing of smart polymers with recognition cavities to extract specific analytes from a mixture of incorporating potential interference. Now I'm talking briefly about this method, which is known as molecular imprinted polymer. So it's a method in which target molecule, functional monomers, and cross-linking monomers are co-polymerized. Major significance or importance of this method is its high specificity, robustness, and selectivity for the target molecule. Also, it has better resistance to high temperature and pressure, higher physical robustness towards acid, bases, and organic solvents. The recognition capacity can be kept for many years at ambient temperature, that is, they have high storage or shelf life. So this is basically the diagrammatic representation of this method. We take the template, which is the drug or anything of our interest. We add monomers to this template, and the functional group of the monomers and the functional group of the template will interact with each other. And then in the presence of cross-linker, they polymerize. After polymerization, this template is removed, and we are left with binding sites which are complementary, recognition and binding sites which are complementary to the template size and its structure. Now, coming to the application of MIPS in different fields, it is being used in various fields. For example, it is used in food industry, pharmaceutical and biomedical analysis, and forensic analysis, and environmental analysis also. So forensic is further subdivided into different fields: forensic toxicology, forensic serology, forensic chemistry, forensic ballistic. I'll take each subdivision one by one with respect to the previous literature. So talking about forensic toxicology under the category of legal illegal drugs, Arifin et al. In 2017 used hair samples to extract diazepam and other benzodiazepines using molecularly imprinted solid phase extraction method. Under cannabinoids, Nestig et al. Developed extraction method by combining molecular imprinted polymer with GCMS to extract tetrahydrocannabinol. Under opioids, Anderson et al. In 1997 extracted semridine by using this method. Under stimulants, Millett and Lai in 1998 used online MISPs for extraction of theophylline drug from serum. Under hallucinogens, Chapius Hugon et al. In 2009 developed extraction method for extracting LSD. Now talking about the subdivision of forensic serology, Ogiso et al. In 2006 detected sequence of double-stranded DNA of interest. Under forensic chemistry, nitroaromatic explosives were detected by Laudel et al. In 2011 by using MIPS as extractor. Under forensic ballistic, Pereira et al. In 2014 used MIPS to eliminate diphenylamine, which is an organic residue of gunpowder. So this is the tabular form of all the templates and the techniques for which has been used for extracting and detecting. These are the drugs. This is the media from which forensic samples were extracted. These are the attributes, that is limit of detection, limit of quantification, and these are the references. So moving forward due to shortage of time. Now talking about the conclusions, we aimed to outline the significance of molecular imprinting in different areas of forensic science. The major drawback of forensic sample is the complexity of sample medium and the low concentration of analyte in the sample, like I discussed earlier also. So MIPS is highly specific, stable, and robust, and it has better features. Sorry.
It was just a reminder, 3 minutes are left.
Yeah, just winding up. Yeah, so I was talking about the specific, its significance, why it is important, because it is highly specific, stable, and strong or robust, and it can be used in forensic science. It is more suitable to be used in forensic science. MIPS-based detection provides a promising alternative to the traditional analytical methods. Also, its application in synthesis needs to be explored and applied to the demanding growth of the forensic science. Now, talking about the future scope, this area is required to be explored further to increase the applicability of this technology in biosensing, extraction, and other fields. There are still a few addressed issues which need solution, like template bleeding, incompatibility with aqueous samples, template limitations. Application of MIPS open bureaus to analyze different kinds of materials with different methods. Also, it plays an important role in the major thrust areas of sciences such as sample preparation, extraction, making biosensors, carrier and drug delivery systems, etc. I would like to thank my esteemed supervisor, Dr. Saurabh Chopra, and co-supervisor, Dr. Deepak Sharma, for their invaluable supervision, support, and tutelage during my work. Also, I wish to express my sincere thanks to the Forensic Corner and NFSU for giving me this opportunity. And that's it. These are the references. Thank you so much.
Thank you, ma'am. I would request our national as well as international scientific panel to ask questions, if any. Okay, ma'am, there's a question for you from the comment section from Utkarsh Gupta. He asks, ma'am, how much is the solvent needed to form spherical MIP?
So solvent depends on the type of polymerization you're using. If you're using bulk polymerization, then you need more solvent. But if you're using precipitation polymerization, then the solvent consumption is less. So that depends on the type of polymerization technique you're using.
Thank you so much, ma'am. Our next presenter is Ms. Shreya Arora, who is an assistant professor from SGT University, Haryana. She will be presenting on the topic to compare the quality and quantity of DNA extracted from lip prints and latent fingerprints present on a glass surface.
Thank you, ma'am. I hope my voice is, audible.
Yes, ma'am, you are audible.
Okay. I hope my screen is visible.
Yes, ma'am, it is visible.
Okay, thank you. So good evening, everyone. I am Shreya Arora, Assistant Professor, SCT University. My topic for today presentation will be, I'll be comparing the quantity and the quality of DNA extracted from lip prints and latent fingerprints from that we have on a glass surface, or you can say on a glass tumblers. Introduction. What we all know what genetic material is. Genetic material of an organism is the substance that contains the information specifying the inherited characteristics of the organism. This information must be stable, capable of being expressed in a cell, undergoing accurate amount of replication, and also getting transmitted in an almost unchanged form from parents to its progeny. Here, there are two forms of, genetic material that we could think of, that is DNA and RNA. DNA is found in almost all sorts of organisms, but RNA could be found in some bacteriophages and viruses, even in some of the plants. So moving on, the DNA that we consider in forensic or in our forensic science, it can be extracted from the bodily fluids that we generally work upon. Bodily fluids can be anything: blood, vaginal secretion, sweat, saliva, anything that you can think of. But touch DNA and transfer DNA is something that we'll be talking more about it in this presentation. So now moving on to the forensic importance of DNA extraction out here. But first, we all know DNA extraction or DNA profiling is something irreplaceable, and it has its application in criminal cases, identification of living or dead, paternity disputes, even linking the suspect with the crime scene, paternity disputes, even endangered species, identification of endangered species, in sexual offenses, and many more. But coming on our main topic, that is touch DNA, or you can say transferred DNA. Many researchers have already shown that variable amounts of DNA can be recovered from the hands of an individual, and even full profiles have been generated from fingerprints, which are deposited on a wide range of substrate. There are various theories that have been put forward. First is keratinocytes. Keratinocytes, or you can say, they are present in the epidermis of our skin. They are, there is a process in our body that is known as keratinization. In this, the cells exhibit steady shrinking of the nucleus and condensation of chromatin as they migrate towards the surface. So when the nucleus have already been degenerated, how we are transferring DNA to the surfaces that we touch. So keratinocyte is something very debatable topic. Then there, the other scientist, Mr. Balogh, M.K. Balogh, probably many of you must have heard. He suggested that there are some nucleocyte
Excuse
cells
me,
that act.
Yes. I think your slides are not moving.
Okay. I think.
Yes, now it is moving.
Okay, I think there is some issue with my slideshare. I'll probably be going this way only. Thank you so much for letting me know.
Yes,
now, moving on. So I was telling you about Mr. Balogh, that he suggested that there are few nucleated cells that are being transferred to the hands from further parts of the body, that is being sloughed off from the sebaceous glands or the sweat tubes. There is another theory that has been put forward and is quite debatable, cell-free nucleic acid, that is CNAs. They say that, since you discover or you recovered many or the contributing DNA from the touched surfaces, there are various level of CNAs that has been reported in serum and in the urine sample. And it could be possible that this type of DNA is what you have in the touched surfaces. So our human body shed approximately 4 lakh cells daily, and in the there are various factors that affect this shedder status that may differ from person to person: the washing of hands, personal habit, type of contact, substrate perspiration. Even lip prints have grooves and this frictional ridges, which are actually do not have those. They actually they do not have any sebaceous glands or sweat glands there on our lips. So what could be the DNA extraction protocol to be followed for lip extraction or this DNA extraction from the lip prints? It's basically the buccal cells that you talk about in lips. Yes?
3 minutes left. Please wind up.
Okay. Okay. The aim and objective would be, to isolate the DNA and compare the quantity and quality of the latent fingerprints and lip prints. So the sample collection was basically done from the glass tumblers. This was basically used with the help of the swabbing, the wet and dry technique, where SDS was used, 2% of SDS solution. The swabbing, for the swabbing part, the DNA extraction, basic organic extraction with minimum uses of phenol. Further, we went through the UV spectrophotometer analysis, where 260 by 280 ratios were compared. Also, the DNA extracted concentration, and then the final process, that is DNA profiling. The results and discussion out here will be these two graphs actually shows the concentration differences that from the latent fingerprint and lip prints. Here you can actually see the graph has much more concentration in case of lip prints as compared to fingerprints. Similarly, when you talk about 260 by 2HD ratio, that will actually help us know, the quality of the DNA that is being accessed out here. We get much better results in case of lip prints as compared to fingerprints. The quantity extracted from the latent fingerprint, it ranges from 0.2 to 125 micrograms per liter. Conclusion will be after extracting of DNA, that study deduced that the considerable amount of DNA can be isolated from glass tumblers from both lip prints and latent fingerprints. The average quantity is being compared, and it was much better in case of lip prints. Also, further DNA extraction was done. DNA extraction was already done. DNA profiling was done using PCR and SCR analysis, and more number of profiles and more number of better profiles were generated in case of lip prints and fingerprint as compared to fingerprints. So these were some references. Thank you.
Thank you so much, ma'am. So any questions from our scientific panel? So, ma'am, we have a question from Utkarsh Gupta.
Yes.
How much should be the DNA concentration for sequencing?
There are various studies being has been done. Earlier it was 100 picograms. Now it has been shifted to approximately 1 picogram of DNA that can be extracted. Even the kits earlier were approximately 0.1 nanogram that they used to work upon, but things have improved since the technology has been improving, and you need approximately 1 picogram and even approximately 200 cells that you need actually to have to generate a DNA profile.
Thank you so much, ma'am. Our next presenter is Mr. Vikas Razdan, who is a cyber forensics expert from In-Service India. He will be presenting on the topic chip-off technology in mobile forensics.
Very good evening. Good evening to all jury members. My greetings to guest participants and the event hosting team. My topic for the today is chip-off technology in mobile forensics. And if we talk of this mobile phone, it has become a part of our life. And the mobile phone, these devices know us more better than I'm audible.
Yes, sir. Yes.
I'm audible. You are able to see my full screen. Can you just confirm?
Yes, sir. Yes, sir. Your presentation is visible.
Yeah. If we talk of the mobile devices, what information we can get from it, as far as the information which can help the investigating agencies to crack the crime. A cell phone has application and communication data, multimedia files are there, internet history is there, emails are there, and location data. And these evidences, these information from the mobile can help the investigators with leads and can help them to reach to any conclusion and crack the crime. And if we find a damaged mobile in worse condition at the crime scene, then it's a challenge for the investigating agency that how the data can be extracted, that how these call details, call history, text, GPS positions can be, even the MAC address or the EMI number can be extracted from the mobile. Now we have various types of extraction. If we talk of the extraction, there is logical extraction, there is backup extraction, there is file system extraction, and JTAG is there. But when all these type of extractions fail, when these all types of extraction fail, then we rely on the chip of technology only. And this is the latest technology that is it being used by the cyber forensic investigators. And if we talk of the mobile phone, it has an internal memory, it has a SIM card there, it has external memory. We call it as a small chip, we call it as micro card, or it's the small card that holds, that is in GBs, like 128 GB or 256 GB, and external it can be added to the mobile. But when a device is found in damaged condition, these SIM card and the external memory card, they can be isolated. But what about the information that can be obtained from the mobile phone that is in damaged condition? We have the RAM, ROM memory, flash memory, NAND memory, in which all these information can be gathered. If we go for this chip of technology, if we talk of the chip of technology, it's the latest technique used by the cyber forensic investigators. We usually take off the chip, which contains all this information, and using the opening, it's the first step. We go for the opening of the damaged mobile phone. Various steps are there. We get the mobile phone, we open it, and we the third step, as you see, that we take out the we find out where the information, where the chip is, NAND chip is there, so that we can perform forensics on that. If we go, the first step in this is chip unsoldering. It's very complicated. It's very we have to use various techniques. We use rework stations. We have different types of rework stations. We keep that chip in the rework station, and using the temperature, where the temperature is kept that is advisable. It's 240 centigrade, and hot air is blown, and you can see that using blades, we take off these chips out, and the compound. You can see the video. I am playing the video. I have made a video for you all. You can see that how this chip is taken out, and the compound is applied on the chip, and all these it's cleaned very smoothly, and all the pins are there. And now we have other device that is called smartphone kit adapters, and these smartphone kit adapters, they play an important role, and they vary from phone to phone, and they vary from chip to chip. It depends on the how many pins we have in a chip, and as per the requirement, we get that smartphone kit adapters. And once the chip is placed in these smartphone kit adapters, a communication is set up between the computer using the cables. We set up the communication between the chip and the computer. And once the communication is established, and using the proper softwares, you can see that it has diagnosed that once the adapter is connected to the computer, using the softwares, we have the softwares, various softwares.
Sir, 3 minutes left.
Okay, ma'am. Okay. Welcome software is the Exom is there, Autospy is there, but I'm here using the oxygen forensic extractor. You can see once the software is being run, that what information we are getting: application usage, cell towers, cookies, emails, passwords, and SMS, and even the device info we are getting, and the chats, call logs are getting, and these all information play a very important role to the investigator to help them to say that if at the crime scene what things have happened. And as I said, that the smartphones have become the part of our life. They know us more, and from these analysis of these all applications, the emails, the towers, the locations, the exit data, the EMI number, and the social media accounts, we can reach to any conclusion. Thank you very much. Any questions, please?
Thank you, sir. I would request the scientific panel to ask questions. Okay, sir, we have a question for you from the comment section from Utkarsh Gupta. He says, so what is the main objective of chip-off techniques?
Chip-off techniques means it's to just get the information from the mobile, from the damaged mobile. If the mobile is in damaged condition, that we are not able to open it, and if it's find out in water, he might be aware that if he goes to the news and the NI had taken the mobile from the sea and the laptop from the sea during the one of the case in Bombay, and they had taken all the information from that. And second case is that NI had taken the Pulwama attack was there, and they have taken the damaged mobile, and they had been using this technology in our country also. It is just to extract the information, whatever the information is there in the chip, and that can help us to the investigators to crack the crime. that we can lead to any conclusion. We can get some artifacts from that information obtained from the device.
Okay, sir, thank you. Another question comes from Mr. Pawar. He says, in case the full mobile is damaged, how would you recover the data?
Yeah, as I said that we have various extraction, logical extraction is there, backup extraction is there, file. When all these extractions fail, then we see the condition of the mobile. We see where the that chip is located. We go through the architecture of that mobile. We go through that chip, and we use this technology. We use the backup station. We use the environment to take the chip, and then we use the smartphone kit adapters. Then communication is set up, and the same way the information is extracted. It's the technology that is usually used for the damaged phones that are no more accessible, that they don't get on, or they don't get when USB is inserted, or any software they might have washed out. It doesn't work there. The chip of technology is that removing the memory that we have, a NAND memory, flash memory, we're removing that memory from the mobile PCB, printed circuit board, then we operate on that. It's that technology. It's the latest technology that is being used by the forensic investigators.
Thank you, sir. Another question is from Sneha. Does the system work to successfully retrieve data even from iOS version Apple phones? Because I believe they are usually encrypted. So how reliable and useful is this technique?
As far as the, if we go through the mobile architecture, there are different, one is the ROM is there, RAM is there. You can see that, I will show you that in the third slide, you might see that if we go through this, we have RAM, ROM, flash memory is used to store a mobile phone operating system. And usually when we use the encryption, or when we use any passcode, that is usually in the ROM memory, and we are not working on the ROM memory. We bypass that. We are working on the NAND flash that is used to store information. And if some information is encrypted, we have the softwares. We have the softwares that can jailbreaking is there. He or she might search that jailbreaking is there. And easily an access to the iOS phones can be done if it's in encrypted place. Also, we have the softwares that investigating agencies can go through, use those softwares and retrieve the data.
Okay, thank you so much, sir. Our next presenter is Dr. Vinita Saini, who is the professor at SDT University, India. She will be presenting on the topic Forensic Significance of Sexual Dimorphism and Population Variation in Discriminant Function Analysis in Mandibular Variables.
Okay, thank you, Nandini.
Thank you, ma'am.
Is it visible?
Yes, ma'am, it is visible.
Okay, thank you. So, good evening all. I am Dr. Vinita Saini from the Department of Forensic Science, Faculty of Science, SDT University, Gurgaon. This topic is basically related to my PhD work. The mandible is considered one of the best skeletal part to assess sexual dimorphism as well as population variation. This is basically due to developmental stages, rate of growth, time duration, as well as masticatory forces vary between the both sexes. And it is also vary between the population groups. Further, the continuous remodeling due to chewing forces, aging, and occlusal stratus make this bone reliable as well as important for sexual dimorphism. In mandible, sexual dimorphism even present at the age of 9 years, but it is mostly in size, not in the shape. In this study, we have conducted discriminant function analysis, which is used to determine which variable discriminate between two or more naturally occurring groups. Means here we have used two groups, that is male and female. For correct estimation of sex, each population group needs its own standards. So why sex estimation is much important? First reason is correct sex estimation is automatically eliminates half of the population from consideration. Secondly, methods of age as well as stature estimation are also dependent on it. When we conduct personal identification, we need craniofacial reconstruction. And for craniofacial reconstruction also, we need correct sex estimation. Reliability of sex estimation from mandible depends on following factor, that is skeletal assemblage and exposure condition, whether we have found complete mandible or fragmented mandible, what was the age of the subject, whether it was a child, whether it was a adult or old individual, intrinsic variability, the method we have applied, that is metric, visual, or highly sophisticated geometric, morphometric, the specific population being examined, as well as the sample we have included in our discriminant analysis. Aims on objective of the study. First is to estimate the sex using discriminant function analysis of mandibular variable. For this study, I have included forensic sample as well as anatomical sample, which was collected from the GSVM Medical College and BHU. So a total of 245 males and 140 females were included in the study with the age range of 18 to 70 years. The variable included in the study were bicondylar breadth, bicoronoid breadth, chin height, bimental breadth, bigonial breadth, mandibular body length, gonion-gnathion length, condylar height, projective ramus height, minimum ramus breadth, condylar height, maximum ramus breadth, as well as mandibular body height. This is the gonion-gnathion length, maximum mandibular length, condylar length, and condylar breadth. All measurements were taken with the help of sliding caliper in millimeter, and average value is used for the analysis. SPSS 28 trial version was used. We conducted descriptive analysis as well as discriminant analysis. In discriminant analysis, stepwise analysis as well as direct analysis was used, and various functions were developed. Additionally, we have tried to include sexual dimorphic index using the following this formula. This table shows the descriptive analysis for all 17 variables included in the study. Here we can see that only condylar breadth was found to be non-significant, and all other variables are highly significant. The highest sexual dimorphic index was shown by maximum ramus height, and followed by gonion-gnathion length and maximum mandibular length. Here we can see this. And this is
3 minutes are left.
Okay, Nandini, I am concluding. This table shows the sexing accuracy by single variables. In stepwise analysis, 5 variables were selected. This table shows the discriminant function analysis. The highest classification accuracy by stepwise analysis was 81.1%. And in direct, when we tried direct discriminant analysis, in function 5, here we can see that 6 variables were included that provided 84.7% maximum classification accuracy. So in conclusion, highest classification accuracy was achieved by using 6 variables. Best variable was gonion-gnathion length and provided 79.5% classification accuracy. And we must always use large sample size in our study, because when we used large sample size, confidence, reliability, and precision increases, and uncertainty, unreliability decreases. Okay. Thank you. Any question?
Thank you so much, ma'am. I would like to ask our scientific panel if they have any questions they can ask. Michael sir, Robert sir.
It was an excellent presentation. Thank you very much.
Sir. Thank you, sir.
Okay, ma'am, we have a question for you from
Yes,
the
ma'am.
YouTube from Sneha. Would this result show ethnic or regional differences? I understand that this is specific for North India, but I'm just curious.
Yes, in North India, this is specifically for Uttar Pradesh population, which I have developed discriminant function for population of Uttar Pradesh specifically. In North India, also there is also we can say population variation. So we must use in the very beginning. I have said that each population group needs its own standards. So in North India also there are various population groups, and each group needs its own standards.
Okay, thank you so much, ma'am. Now, I would like to call our next presenter, who is Miss Shambhavi Pandey, who is a student from SGT University, India. She will be presenting on the topic, an analysis of sex estimation from handwriting, a review.
Thank you, ma'am. Am I audible?
Yes, and your presentation is visible.
Okay, ma'am. Thank you, ma'am. Good evening to all. I'm Shambhavi Pandey, student of BSc Forensic Science in SGT University, Gurugram. Here I'm presenting on topic an analysis of sex estimation from handwriting, a review. First of all, we talk about what is handwriting. So handwriting is a complex neuromuscular task. We simply mean that our brain, spine, and hand and arm muscles work together to make us write on any surface. It can be both printing or cursive style and can be separated from formal calligraphy. Next is graphology. It is the examination of physical characteristics and pattern of handwriting to identify the writer or of any handwriting sample. Each individual handwriting is unique and has the unique characteristics feature in their handwriting. Sex of an individual is additionally play a significant role in the arrangement of handwriting. There are few of them literatures which I have examined, like Kumar and Saran. They performed a factual investigation of characteristics element in the handwriting sample and found that there is a difference in handwriting of male and female. They found that there is a counters in vowel, delicate hook and curves, consistency in size, round spots over the letters, etc. in female handwriting, while male handwriting has little counters in letter, irregularity in their size, straight line and sharp ends and corners. Next is Upadhyay S. et al. They proposed that out of 27 characteristics of handwriting for which Z-test was performed, and there were 7 highlights, that is dot over I, straight down stroke formation of letter S, angle of crossbar on alphabet F and T, loop in stem of letter D, etc., said to be the 6 recognizing characteristics which may help in the identification of sex of the author. Next is Nader and Muhammad that recommended handwriting can in any case tell whether the author has the feminine or masculine characteristics. They consolidated all the highlight to gain the normal exhibition of feature 1 to feature 26 on English and Arabic content. They found that the female handwriting has more significant variation with male. Young and Term et al. Propose that there is a novel neural signature of 6 differences in a sign of human behavior and highlights the importance of considering sex as a factor in scientific research and clinical application involving handwriting. Here is on the basis of literature which I have examined, I conclude that there are various factors that cause the handwriting difference in male and female. That is like on neural basis and their day-to-day habits, embellishment, growth factor, and their writing skills. Because of the various methodologies of male and women towards life and due to the various qualities in life, they build different habits in handwriting. And handwriting is also called the brain writing. So because in the difference, their thinking and hormonal inclusion, writing of male and female varies. It's seen that the female handwriting perform better as compared to male in fine motor ability. It was also seen that the females are more affected by the beautification and decoration of letters, so they produce handwriting in which designed letters, while male writing contains cramp and little counters in vowels, irregularity in size, harsh and occupied shows that their attributes by not giving a lot of significance to the letter decoration and focus on the completion of writing with sensible neatness. We also seen that even twins who have a similar appearance and offer hereditary qualities do not have the similar handwriting. It is because of the way that one grows up and learn their first language, the force and the method of forming words to make a one of kind of way of writing for every single individual. We also seen that there is a difference in writing skill of male and female, like the text length, vocabulary, writing fluency, text quality, and self-adequacy. Sex estimation from handwriting could be useful in predicting the sexual orientation of the writer and just help in the recognizing the culprit from the suspect and introducing solid
3
proof.
minutes left.
Okay, ma'am. Solid proof in the court of law. So here is references. Thank you so much.
Thank you so much. Now I would like to ask our scientific panel if they have any questions. Eugenio, sir. Rajesh, sir.
Yeah, I have a question. Do you think that male and female or masculine and feminine qualities can cloud it if you isolate them from being male or female? And then also, where does education come in when you start talking about spelling and text length and stuff like that?
Yes, sir. I think that their education matter, and like the text length, text quality, it matter to study about their handwriting differences.
Any further questions?
Okay, so moving on to our next presenter, who is Miss Van Mohammed Tawfiq, who's a student from University Saints, Malaysia. She will be presenting on the topic preliminary study on classification of individuals using handwritten numeral with geometric morphometric technique.
Thank you, ma'am. Is that my voice clear right now?
Yes, ma'am.
Is my slide shown on the screen right now?
Yes, ma'am.
Okay, let me do the slideshow. Is appear right now? Hello?
Yes, ma'am.
Okay, can I proceed? Okay. Good day, everyone. My name is Wanurul Shafawani. I am a PhD candidate from the Forensic Science Program, School of Health Sciences, University Science Malaysia. Today, I will present about the preliminary study on the classification of individuals by using handwritten numeral with geometric morphometric techniques. Fingerprint and DNA evidence has long been used in the forensic investigation for individual identification. However, numeral handwriting also can be used as a crucial evidence in the court of the law, along with the fingerprint and DNA evidence for the individual identification. Numeral handwriting is a writing system that uses a single or a combination of numeral digits to express the number of given range by using the writing matrix. And it is believed that handwriting of individuals are influenced by many factors, including their ethnicity, gender, handedness, education, or age as well. And this study was focused on the use of geometric morphometric technique in order to identify which handwritten numeral that have the potential for discrimination between individuals. And GMM technique also was applied to analyze and discriminate different individuals based on their numeral handwriting. By introducing this GMM technique into the field, Probably forensic document examiner could apply this technique in order to determine the individual by using handwritten numerals, which could be found as an evidence in document from investigation that involves several people with different criteria. Handwritten 1, 6, 8, and 9 were collected from 10 Malaysian students from University Science Malaysia, Kelantan, Malaysia. All these participants were asked to write numeral of 1, 6, and 8, and 9 on A4 white paper for 10 times within 10 separate rows. All these numerals were chosen based on the previous study by T 2019, as they successfully differentiate the individuals by using fully manual methods based on their length, width, and height measurement before statistical analysis, which are ANOVA and Kruskal-Wallis tests. However, there were no specific criteria to be focused on this study. And we also want several software to extract and evaluate the handwritten numeral samples. For example, TPSRT, TPSD2 software, and Morpho-J software. All the acquired samples were then converted into digital image prior to image processing by following the scheme that's shown on the screen right now. There are 3 important stages. The first stage were pre-processing stage. This stage was performed using filters that are available in the Adobe Photoshop in order to enhance the brightness, the contrast, to remove some extra pixels and noise reduction. And then we proceed with digitizing and landmarking stage. The digital image were then inserted into TPSRT for digitizing purpose, and then inserted into TPSD software for landmark positioning prior to the principal component analysis, which also known as PCA in the Morpho-J. Principal component analysis was chosen from the variation menu in the Morpho-J as shown in the figure 1. Principal component analysis was performed in order to identify and then explore the morphological pattern that can be used to discriminate the handwritten numerals 1, 6, 8, and 9 between different individuals. PCA score plot was generated after we run the shape analysis derived from the covariance matrix that produced by the dataset and show the clustering of data with the same characteristic. And then result and discussion. The handwritten numeral 1, 6, 8, and 9, which call it. Oh, sorry, ma'am.
3 minutes left.
3 minutes remaining.
Okay, okay, I move forward. The handwritten numerals 1, 6, and 9, which collected from 10 individuals, did show some distinct clustering and some degree of dissimilarity of character between individuals. And the score plot of PCA revealed that numeral 6 was the only one numeral that exhibited distinct clustering as shown in the figure 2. Besides, based on the PCA score plot, there was some cluster that overlapping between different individuals for numeral 8 and 9 as shown in the figure 3A and 3B. This is because likely due to the significant similarity in the general shape of numeral between different individuals and the presence of the feature variation within numeral 8 and 9. However, only numeral 1 that did not display any distinct clustering within individuals, as we can see in the figure 4. Probably this could be sample from these 10 individuals has significant similarity in the general shape of numeral 1, as we all know that numeral 1 is likely to construct with one stroke character. Besides, it might be due to inadequate numbers of landmarks, or probably inappropriate positioning of the landmark when we do the digitized handwritten numerals. Or yeah, probably simply because the handwritten numeral 1 sample will indeed appear similar in characters. The conclusion, geometric morphometry can be employed for discrimination of individuals by using the handwritten numerals. However, increasing the number of samples for GMM analysis will be more advantageous since any undesirable sample could be eliminated from pre-screening results in order to enhance the data accuracy. All these findings should be followed by the prosecutor ANOVA in order to determine the significant difference between individuals. Aside from that, geometric morphometry can be complemented with other machine learning techniques for comprehensive data analysis to overcome the shortcoming of the GMM technique. Finally, probably other criteria or characteristics such as genders, ethnicity, education, or handedness can be introduced and focused in the future for better assessment. This is my reference. That's all for me. Thank you.
Thank you so much, ma'am. So, do we have any questions for her from our scientific?
I like it.
Okay, so now I would like to call our next presenter, Miss.
Okay, thank you, ma'am.
Okay, thank you so much, ma'am. Our next presenter is Ms. Vidushi Saraswat, who is a student from NFSU India. She will be presenting on the topic determination of root cause of victimism and its inducing agents.
Hello, am I audible?
Yes, you are.
Is my screen visible?
Yes, Vidushi, go ahead.
So I extend my greetings to all the present dignitaries and the participants. My topic for today's discussion is determination of root cause of victimism and its inducing agents for the fulfillment of my universal certification in forensic psychology and criminal profiling. Now, first of all, talking about forensic psychology. It can be described as an integrated field dealing with science and law with the psychological interventions that help in the forensic investigations. Although we all know forensic psychology can be classified into two broad groups, that is victim psychology and criminal psychology. Criminal psychology includes characterization, categorization, as well as offender profiling. But the victim psychology is a field which is underdeveloped and what I want to talk about. So, in experimental mindsets, associative links are formed between the victim and the negative event. That is, the people who has experienced any trauma or has been a witness to any kind of exposure to such events may judge such situations on the basis of their experience rather than thinking logically and rationally. There are 6 broad classifications of victim given, starting from the innocent victim, which can be classified as a child, again continuing, victim being slightly guilty, victim more guiltier than the offender, and lastly to the imaginary victim. Imaginary victim is the main topic of my discussion. Also, another advantage of these determination and categorizations of the trends, traits, and the inducing agents would be a prevention and control of an individual to fall into self-victimization and alternatively prevent them from being the misleads into forensic investigations. Literature review. Victimhood can be attributed to many factors such as exposure to some traumatic event, undeserved harm, vulnerability of an individual. Vulnerability being defined as age, gender of an individual, and also the disabilities, listing into the many more factors. So, blaming categorizations very well explain the role of gender in type of victim blaming. For example, in behavioral blame, we take into account the variable victim factor that can be explained as victim action. Also, in characterological blame, we take into account the stable victim factor that is, for example, can be explained as a female being naive and being blamed for that. The identity of an individual tends to rely more and more on pseudo-religion of victimhood. That is, people find victimhood as an inspiration when there is a history of ancestral victimhood. For example, in Jewish Holocaust and the massacre of Nanking, the Chinese Americans taking the victimhood of their ancestors as a peculiar source of pride. Although all the aspects, traits, triggers, and or inducing agents have been discussed in the previous studies, but they are all about the post-traumatic event and the exposure after the exposure. What I want to talk about is pre-exposure to the traumatic event here. Research hypothesis and objective. The hypothesis proposed with this article is that onset of victimism follows a trend. A trend that is here, that is the more need of sympathy over empathy, more need of over attention over recognition, small grief changing into prolonged grief, antisocial behavior, egocentricity, aggression, and anti-optimistic behavior. So now, here the term comes pessimism. We can also talk about the similar term that is narcissism. Narcissism, as the term says, is an inflated sense of self-importance. That gives a person a false idea of the self. So here is a relationship established between the pity and the pleasure. The person enjoys the pity given by the others. So there is a syndrome known as narcissistic victim syndrome. So the primary level of victimism starts at a very young age, that is the most vulnerable age, most attributing factor being the vulnerability of the child. This being the pandemic condition of victimization. The victimization is classified into 3 classes, that is pandemic, acute, and extreme. While talking about pandemic, it can be explained as sibling fights, for example, 2 siblings fighting with each other and blaming each other and victimizing the self. Acute condition can be explained as being a victim or being a witness to an assault. Extreme, experiencing or being a witness to a homicide. It is proposed here that the young adults, specifically teenagers, fall prey to this victimization more as compared to the adults. Also, the middle-aged adults show more inclination towards just word belief. Sorry?
Please wind up. 3 minutes left.
Okay. The types of inducers I have listed are interdependent upon each other. More specifically, I want to talk about the historical inducers that give a peculiar source of pride, and that is the questionable section of the victimhood. Methodology. The methodology I have employed here is hypothetical vigilate methodology, in which a hypothetical situation has been given to the subject target groups and their logical and rational perspectives has been recorded and analyzed for the categorizations and characterizations. The scope. This research will mainly help in psychological inventions by identification of vicarious victims, that is, identifying self as a victim for the sufferings of the other or eliminating a mislead from the investigation. Thus, this research will indirectly become a tool of forensic psychology in legal prosecutions. Also, clinical psychology, that is, can be an extended area for providing comprehensive psychological support to individuals developing victimism. Findings and conclusion. The victim resistance is directly linked to victim gender, shows a direct linkage to the victim blaming. Also, depending upon the homosexuality and heterosexuality of an individual, mostly blamed victims are the submissive male victims. Young adults show higher tendency of developing victimized persona. In that case also, the females are more prone to that because of the cultural and social conditions. The experience of some past event or trauma hinders with the rational and logical thinking about the current present scenario. Pandemic victimization can be taken as the basic building block that triggers the vulnerable children. In historical inducer perspective, victimhood is questionably taken as an inspiration. Now I will end my presentation here. These are the references. Thank you.
Thank you so much. I would request our scientific panel to please type on their questions because it is taking so much time. Okay, we have a question for you, Vidushi, from my YouTube. Does your study can help differentiate between sociopath and psychopath?
It is more concerned with identifying the actual victim and the fake victims. As you know, also in forensic psychology, what we do is creating the profiles of the criminal, on which that helps in forensic identification. So although narcissism and pessimism can be taken in psychopaths and all these categories, but this is more prone to identification and treatment of such people.
Okay, thank you so much. Moving on towards our next presenter, who is Dr. Abdeli Sali, who is a forensic odontologist from Forensic Odontology Institution, Sudan. He will be presenting on the topic of human canines as a forensic tool for gender determination.
Hello?
Yes, sir. Sir, I think you have muted yourself.
Hello?
Yes, sir.
Oh, sorry. Am I sharing the screen now? There we go.
Yeah, I can see,
Is it
sir.
clear?
Can you rotate? Can you
Okay,
rotate your screen?
Okay. So I'm gonna use my cell phone. I do have some technical issues with my laptop. Apologize for that.
Sir, shall we present?
Afternoon. Okay, afternoon, ladies and gentlemen. Today we're gonna talk about the human canines and how we can use the human canines as a forensic tool for gender determination. I'm sharing many theories of mine. We all know that different species have different biological structure and variation in the size and the shape. Either human beings, either animals, either plants. We all have the different biological variations between the males and females. And what I mean by the gender, it's quite different from sex. Gender is more psychological and more religious based than sex, which is sex is biological. Okay, production forensic odontology is mainly used in the identification of the deceased by using their profiling their age, gender, and ethnicity, either using their scalp or dental structure. In the catastrophic disaster or the mass fatality incident disaster victim identification, the forensic dentistry might be used for age estimation, gender estimation, and profiling of the deceased individual. The primary methods or the scientific methods of human identifications are the fingerprint, the DNA, the dentition, and the medical record. Sometimes it might not be applicable, so the dentition may be quite helpful in such cases, especially if there is fragmented or incinerated remains. There is different studies that have been used in the literature by using the maxillary incisor, the lateral incisor, and the canine, the first molar. The canine anatomically is the third tooth from the lower or left quadrant. We do have 4 canines, 2 upper and 2 lower, 2 upper and 2 lower. Why the canine? Especially the canine. The canine is quite unique, one of the most anterior unique teeth. Quite resilient tooth. It has minimal effect of adhesion during the mastication or the tooth brushing. It's quite healthy and less affected with the caries and periodontal diseases or the gum diseases. And that's why we can use it as a key tool for dental identification. In this research methodology was quite simple. is by measuring the maximum width of the distal part of the canines, the upper and lower canines, by using the digital caliper of 50 males and females of sample of the South African population in Cape Town. Digital caliper was used, field caliper, with a quite simple formula by graph, the xm xm over xf minus 1 multiply 100, while the xm represents the medial distal width in males, the xf represented the medial distal width in females. Minus 1 multiply 100. This is the formula to estimate the percentage of the gender probability determination. The result shows that we can estimate the lower left mandibular canine with a percentage of 72 gender determination probability, with a 69% of the lower with the lower left mandibular premolar, mandibular canine, with the 60%, the upper right canine does not show any significant feature. The upper left canine shows almost 66% gender determination probability. In the discussion, different
Sir, 3 minutes left.
Okay, different studies in the literature scored almost 70 to 78%, as you can see, from Turkey, from Lebanon, from from South Africa as well, from Spain and from Sudan. The average was 70, 75, 72, and even until 84. We can also use the gender determination probability using the canine width in archaeology or bioarchaeology. In Croatia, in 2007, there was excavation of 86 skulls. Odontometric analysis were used in Italy as well. 2015 applied odontometric analysis in 149 skulls remains. In Portugal, of 115 remains, they use odontometric analysis. In South Africa, as well, one of the recent studies, sorry, we didn't write the year, but I think it was 2013, 14, about 115 human skeletons. The gender determination probability was 75%. This is odontology in bioarchaeology, so it's a combination that can be used different basis or different topics. Thank you.
Thank you so much, sir. Do we have any
Sorry
questions
for
from the panel?
any, yeah.
Hello, am I audible?
Yes, ma'am.
Hello.
Okay, there are few questions from my side. Like, according to you, which are various detrimental effects of habits that interfere this canine dimorphism?
Say again, sorry, I can't hear you. The canine width?
Which are detrimental effects of various habits that interfere in this gender dimorphism related to canines?
You're talking about the habits, if there is any habit of
Yes.
chewing or persisting. Yes, I'm using the medial distal width. I'm not using the length. I'm using the width of the canine. So it's not going to be highly affected unless it's behind the midline of the canine. So I'm just using the width. If it's the length, it would be quite complicated. So I'm just using the width. of the canine, not the link. But it might affect if there is any bruxism or clean sheet. So, and it was one of the exclusion criteria that if there is a lesion or wearing of the canine or dental structure. Good question.
Okay. Is there any role of regressive changes like accretion?
Sorry?
Is there any effect of accretion?
Accretion?
Yes.
Yes, it might affect. Accretion, it might affect. As I mentioned, it was one of the exclusion criteria in this many thesis. Exclusion of any accretion. It might affect, yes. It might affect the length more than the width. It might affect the length more than the width. So I'm measuring the width, not the length. But it's gonna affect if there is accretion or persistence, as I mentioned.
Okay. Thank you.
Okay.
Thank you, ma'am.
Thank you. Thank you. Thank you. Thank you
Sir,
for
I
this.
have one more question for you. Hello?
I think he left the meeting. We should carry on.
Okay, moving on to the next presenter. Our next presenter is Dr. Abhishek Kumar, who is a student from NFSU, India. He will be presenting on the topic crime mapping, constructing a safer community. Sir, are you there?
You're not audible.
I think sir is having some network issues. He can continue with the next presentation.
Okay, sure. The next presenter is Miss Mona Sharon, who is a student from Jain Deemed to be University, Bengaluru, India. She will be presenting on the topic age determination of blood-stained fingerprints using hyperspectral imaging.
Good evening, everybody. Am I audible?
Yes, ma'am.
Yes. Okay. So today I'm going to be presenting to you about age determination of blood-stained fingerprints using hyperspectral imaging, which is a literature review that I have done. So moving on, I'd like to quickly, the slides aren't moving. Yeah. I would like to quickly give you a brief about what is hyperspectral imaging. It is a digital imaging technology of two spectroscopic methods, which is using hyperspectral imaging spectroscopy and fluorescence spectroscopy. And the main principle that it works on is collecting images as a function of a wavelength. And what it does is it provides an individualized spectrum for each pixel on the image of the image. And using that, we will be able to tell what are the compositions and the components or the unique features of that picture on the image. So for example, in this, there is a picture here, and this has a unique spectral signature, from which we can tell many of its features. So moving on here, I would like to just give you a quick brief about the kind of setup that was used for these research papers. It was done by a group of people, research scholars, the same people who kept on researching further on these things. So here we see that they've used a liquid crystal tunable filter and a CMOS camera and a 230 watts solid state plasma light source. This was used as version 1, and an improvised version of this was used as version 2. And on your right, you see that you can see the actual setup of how HSI is normally set up. They've also used a custom software to control the image capture and also the LCTF, and they've used a modified version of Kullbacher-Monk's equation. And moving on to paper 1, quickly, I would like to tell you that they've done on 4 trials. They've done using 4 trials. One was for using undiluted blood stain on dark colored substrates, which are very similar to the color of blood. Then they've also used 9 substances. They found out 9 substances which can be commonly confused as blood. and they've deposited on paper, on a normal photocopier paper like the printer paper, and then they've used a series of diluted blood and deposited that on white filter paper and also on red tissue paper. And the fourth trial was done using a blind trial, which on a red t-shirt which was covered with blood stains, and this t-shirt was given by the Metropolitan Police of London. And then the results of this paper, research paper was that they were able to determine the shape of the stain clearly, and they were able to identify that the different spectral peaks for each of these 9 substances, and they found that each of them had various different peaks through, and bloodstain had a very different one, which was the 415 nanometers, was also called a Soret peak. And then they also were able to determine up to 512 folds of dilutions on white filter paper, and up to 32 fold dilutions on red tissue paper. And on this red t-shirt, they were able to find 9 blood stains, out of which 1 was very faint, and that they were able to identify and detect using the HSI. Now, on moving on to paper number 2, they've done again on 4 trials, where one was using dilutions, and here in the dilution stage, they've done using where they've compared HSI version 1 with a DSLR, and in stage B, they've used HSI version 1 and compared it with HSI version 2. And in third trial, they studied about the different contaminants, which was similar to the previous one, but here they deposited on a white ceramic tile. And then the fourth trial was done wherein they tried to deposit a thick and thin blood on a white ceramic tile, and they aged it in an environmental chamber for the 6 months to check if the HSI can detect that. And the results were very promising, that they found out that they could identify the whole ridge detail up to the 6th depletion, and partial ridge detail up to 9th depletion, and they could detect the bloodstain up to the 12th depletion. A DSLR could find out the whole ridge details up to 3rd fold. HSI could do it, find out and detect up to 5th fold. Partial ridge detail was detected up to 5th fold by a DSLR, and 20th fold by a HSI version 1. And HSI version 1 could detect up to 100 folds of 5 stains of blood stains without the ridge details. Moving on, the HSI version 2 was found to be more superior and much better because of the improvised devices that they used. It was able to detect up to 20-fold dilutions. And here, and the contaminants—
3 minutes left.
Thank you. They were easily able to distinguish between the contaminants, and the ridge detail that were aged up to 6 months were easily visible using HSI. And moving on to paper number 3, they've also done it on dark tiles, light tiles, black tiles using version 2. They've also done on various porous and non-porous substrates, and ranging from wood, cardboard, cotton, and plastic paper and glasses, and also on pig skins, wherein they use it on, they deposit wet prints and dry prints, and the results also was found out that version 1 works better for white tiles, version 2 works better for dark tiles. And they found out that even the ridge details could be found out even after the fingerprints were depleted using HSI on porous substrates, and on non-porous as well. The results were very promising. And finally, the wet stains on the pig skin were detected up to 1,500 fold dilutions, and dry stains up to 1,000 fold dilutions. Finally, the paper number 4 was that they did it on 3 age estimation intervals of 24 hours, 30 days, and 9 randomly aged bloodstains. And these are the results. So here we see that they were able to clearly and successfully detect the ridge details for all the age periods. However, here they found out that after 14-day period, that the absorption spectrum decreased. And so the colors became more like shades of purple, which was a bit hard and challenging here. So let's see what discussion is. What can HSI do? Basically, it can detect a blood stain because of the specific absorption peak, that is at 415 nanometers. It can also identify the blood stain from other stains and dilute it when, even if it is diluted or depleted. And it can also identify when the surface on different substrates or surfaces, and also the age of the blood stain can be detected or determined, and you can also find out the rich details. Now, moving finally, the limitations is that it does not work on black substrates, it does not work on paper or red or black cotton fabric, and use of the false color to determine ages of the fingerprint after 14 days is a bit challenging. So because of the reduced absorption spectrum. So the suggestions that we can that I have come up and also I found out on the papers were that an improvised HSI setup is required to give a tough challenge for a DSLR. And also that if we could use ultraviolet light, we could use it on a different biological specimens. And further, if we could generate a custom software so that we can export the data to different devices for comparison, I think that would be very efficient. And finally, if we could
time
have,
is up.
Okay, can I read the last line? That if we
will
had
take questions now.
Okay.
Take questions.
Thank you.
Okay, so the question is from Dr. Neelkant sir. He asked, hyperspectral imaging is targeting blood constituents or fingerprint constituents?
Mainly blood constituents because of the spectral peak, that unique spectral peak at 415 nanometers.
Okay, so another question is from Rajveer, sir, is this method applicable for other biological fluids also?
The thing is, I haven't come across a paper where they done it on other fluids, where they've used HSI for other fluids, or even if they've done that, I think I didn't read that much on it. But yeah, there is scope for that. Yes.
Okay, thank you so much, ma'am. Our next presenter is Ms. Priyanka Yadav, who is a research scholar from Maharishi Dayanand University, Rohtak, India. She will be presenting on the topic to study pollen ornamental plants in forensic context.
I'm sorry to interrupt, but actually we have a question from our scientific panel by Eugenio sir for the previous presenter. If Nandini could take it, please.
Yeah, okay. So Mona, we have one more question. How big is the reference sample that you use for the studies?
So actually the thing is I have done a literature review, so I didn't do the research myself.
Okay, thank you.
Thank you, sir.
Priyanka, your slides are visible.
Hello. Am I audible?
Yes.
Okay. Good evening, everyone. My topic is to study pollen morphology of ornamental plants in forensic context. I'm a research scholar from the Maharishi Dayanand University, Rohtak, in Department of Forensic Science. Now, we talk about the palynology that has evolved from the palyno, which means to sprinkle, and pale, which means to dust. The first microscopic study of pollen was done by a British named Grew in 1640 approximately. Palynology was restricted to study of spores and pollen grains earlier, but now it is evaluating as interdisciplinary science and involves study of surviving and extinct polymorphs. Pollen has interactive nature, that's why it has contrasting range of approaches such as forensic palynology, aerobiology, paleopalynology, mesopalynology, limnology study, pollen biology, archaeopalynology, etc. The analysis of polymorphs found in both marine and freshwater environment are included in forensic palynology for legal perspectives. Palynology turns in forensic palynology when it is used to obtain supplementary evidence for legal purpose. Pollen grains are basically produced by the meiosis process, so they are haploid. These represent the sperm of plant world and are male reproductive structures that are produced by the flowering plants, angiosperms and gymnosperms. Pollen grains has two walls, outer cellulosic coat exine that is strengthened by sporopollen, which provide resistance to the decay and protect inner intine, and that's why we use pollen grain as a evidence as it resists decay for a long time, even years. So pollen as a evidence, pollen is being used as calibrating evidence because of microscopic size, resistant to decay, and can easily adhere on various objects of almost every type of surface unnoticed. Pollen is capable referred as golden dust because of its appearance as fine yellow dust. Sometimes it also referred as wonder dust. Pollens are known as nature's fingerprint of plant because of its distinctive morphological features and unique characteristics. Forensically, pollen grains analysis help in police inquiry, narrow down list of suspects, relate them or suspect to the crime scene, determine travel history, pre-mortem fate of a victim, season of crime occurred. Forensic palynologists help in two types of investigation. One is to associate the item linked with the victim, suspect, or crime scenes. I to prove or disprove a suspect alibi. Either he or she was present there or not. And another one is to indicate the geolocation of samples with unknown origin. Forensic palynology can become a powerful tool for solving crime as spotty row. So when we go for experimental work, following are the objectives that I have choose to study forensic palynology, which will cover due to lack of awareness and appropriate database, palynology evidence has been ignored in most of the criminal cases. Even in India, that is a developing country, but developed country use it as a evidence in various approaches in legal investigation. In this, we study the following objectives: to study the forensic value of pollens by description of their morphology on the basis of color, shape, aperture type, sculpture pattern, and pollination method of 20 angiospermic plants comprising the ornamental species. How we collect them? Pollen dust of various plant species along with their anther globe are collected and then preserved in 70% alcohol resist that because it resists bacterial attack on this moist biological material. Slide preparation of pollen samples. Pollen from 20 different flowering plants collected from Herbal Garden of Maastricht University, Botanical Garden of Hisar Agriculture University, Botanical Garden of Gurugram, and Rewari, Haryana, India. Preserved pollen samples are treated with acetylysis and chlorination process as described by the Piquet. Following are the results. When we go through the microscopic study, results clarify that pollen grains exhibit considerable variation in their morphological characters such as in color, shape, aperture, and sculpture. So unique pollen grains found from a crime scene help in legal investigation. In this table, there is a description of the scientific name of plant, their common name, family, color, shape, aperture type, sculpture type, and pollination method. These are given in elaborated way how it vary in their color, shape, aperture, sculpture, and pollination method. So this is another table which also comprise the ornamental plants and pollen grains of these species also vary from boat shape to rounded, cylindrical, circular, spherical, etc. and their aperture size as well as sculpture size. Both show distinct differences in their structures and types.
3 minutes left.
Okay, these are the pictures which I have studied at 100x microscope. Following are A is a picture of flower where I have collected from the pollen samples, and the other one is stained pollen, and C and D belong to acetylized, and E1 is chlorinated. These are the pictures of the pollen morphology of different ornamental plant species. These belongs to different species, and we can see the differences in their morphology. Shape, size also vary at a great extent. When we go for discussion in civil and criminal cases, pollen is used as law enforcement for almost 15 years. It can adhere to any surface and we can characterize different environments. In this study, we found significant differences in the different genera of pollen morphology. Differences are based on their morphological feature like shape, sculpture pattern, and it can be helpful in identifying class at genus level by examining their morphology. Ornamental plants show characteristic differences among pollen morphology and used as identification tool for different rural and urban areas. Pollen retrieval from a crime scene also help in estimating crime timing as pollen are season specific. We can retrieve it from hairs. Indoor ornamental plants are also helping criminal investigation. They exhibit unique pollen spectra in accordance to their texta, and the use and application of forensic pollen studies are relatively new in many developing countries like India. Reasonable factors for pollinological rare application are large geographical area, huge number of plants, general lack of research, and lack of awareness. So there is need of a database to study pollen barcoding to identify the plants at genus level and species level. And there is need of pollen DNA barcoding for identification of pollen up to species level. So more research is needed in pollen genetics to understand the genomics of pollen. Thank you. These are the references. Thank you.
Thank you so much, ma'am. Do we have any questions from the scientific panel? Okay, so one question comes from Dr. Neelkant sir. Ornamental plants mostly consist of self-pollination or closed pollination, where chances of pollen spread is minimal. So what is the probability of encountering pollens in indoor environments?
Actually, indoor environments is not only specific to a room. There are a lot of people that go for their hobbies, like to make a garden against their house, home, and there are several many plants which show cross pollination. Yeah, there is a minimum chance of pollen spread, and in case of self-pollinated plants, but we cannot exclude it as an evidence. There are always some limitation with every evidence. So basically, these are also included in the pollen case. But we can go for the ornamental plants are not only the small plants that go on for self-pollination. There are also many big plants like rose, in which there is not possible that is only in a room. We can see a garden in every house, only even in the houses like in sectors of some cities, there is also some spaces also available for the garden areas. So it is not only, we can not only exclude this pollen evidence because we can go for it only as a self-pollination method. There are also other chances where we can use it as an evidence. It is not solely used as an evidence, it is a collaborative evidence, so that help as a supportive role for crime solving.
Okay, the next question comes from Dr. Hadi ma'am. Is there any other method apart from microscopy to differentiate various species?
Microscopic के अलावा we can go for the DNA barcoding, so we can go to the species level. DNA barcoding of pollen.
Okay, thank you so much.
You're welcome. Thank you.
Moving on to our next presenter, Mr. Tahir Ul Ghani Mir, who is a research scholar from lovely Professional University, Punjab, India. He will be presenting on the topic quality estimation of commercial Kashmiri saffron using UV visible spectrometry and high performance liquid chromatography analysis.
Is my screen visible, ma'am?
Yes, sir.
Okay. Good evening, everyone. My name is Tahir Ul Ghani Mir. I'm a research scholar at Department of Forensic Science, Lovely Professional University. So today I'm going to present my topic called estimation of commercial saffron and using UV spectroscopy and SPLC liquid chromatographic analysis. So this is the part of my project. We are working on detection of adulteration in saffron. So first of all, classification. So saffron, it's obtained from Crocus stivus. Crocus stivus belongs to the family Iridaceae of order Asparagus. So saffron, it is the world's most expensive spice, also called as red gold. It's grown in Iran, Spain, Green, and in India, it's mainly grown in Kashmir. So due to low production, high demand, and expensive labor work, this spice is very high priced, and because of which it's very prone to adulteration. So here is the phytochemistry of saffron. Saffron, it contains many volatile and non-volatile active components. The volatile components are terpenes and esters. Non-volatile components are crocin, picrocrocin, saffron, and crocin. This crocin, it's responsible for color of saffron. Picrocrocin, it's responsible for the bitter taste of saffron, and saffron reduces the aroma of saffron. And this crocin, it is responsible for the smell of the saffron. So here is the ISO grades of ISO 3632. This is the international standard organization. They have classified the saffron in 3 categories based upon their quality. So this ISO 3632, this certifies customers that the saffron they buy is genuine and safe to use. They have classified saffron into grade 1, grade 2, grade 3 based on the amount of the metabolite is present in the saffron. That is picrocrocin, saffron, crocin. Presence of these components in saffron, the higher concentration of these components means the higher grade of the saffron. So here are the adulteration methods usually adopted for adulteration of saffron, like various maize, slick, saffron stamen, safflower, marigold. Also, turmeric is added, safflower, it's colonithisms like saffron, dyed fake saffron. These are some adulteration manners used in adulteration of saffron. So in this study, material and methodology, 24 samples were collected from different geographical locations of Kashmir. And besides these 24 samples, 4 samples were collected from Afghanistan and 2 samples were collected from Iran. And 1 sample was collected from Sigma. Total 31 samples were used in the study. And standards of crocin, picrocrocin, and saffron, they were obtained from Sigma. So to investigate the adulteration of saffron, first we used UV spectroscopy according to the ISO method. So in order to go for the study, first we need to detect a moisture content and floral waste in our sample. So using these equations according to ISO, we were able to determine the moisture content and floral waste in our samples. Next, we went for UV spectroscopy according to ISO, and the measurement of E1% of the saffron extract at 440, 330, and 257 nanometers. Using 1 centimeter per quartz, results were obtained by direct reading of the absorbance, and absorbance obtained from the results were used in this relation to calculate the E1%. And this is the ISO 3632 method to determine to which category the saffron belongs. Next was HPLC analysis. So HPLC analysis, we use a Siemens HPLC equipped with PDA detector. And separation was carried out with injection volume of 20 microliter at flow rate of 1 microliter per minute, and the runtime was for 35 to 45 minutes. The saffronal metabolite was detected at 310 nanometer, and crocin was detected at 440 nanometer. So here are results and discussions. Results. This is the data we obtained from the while calculating the moisture content and floral waste of our different samples. Next, we under UV analysis, this is the data we obtained under UV scan. So in this results, we found that only 3 samples we studied belong to the grade 1, and 4 samples were completely fake. That means they were completely adulterated, and remaining samples they were either they either fall in grade 2 or grade 3 category of saffron. Another this HPLC analysis was conducted to determine the concentration of metabolite like crocin and saffron per mg. Sorry, sir.
Sorry to interrupt you, but only 3 more minutes left, sir.
Okay, sir. So we used HPLC analysis to determine the concentration of crocin and saffron in our samples. So standards were used, and the data was obtained for analysis. So conclusion in our analysis, after analysis, we found that only 3 samples belong to the grade 1, and 3 samples were completely adulterated, and remaining 18 samples were either grade 1 or grade 2 or grade 3. So due to because this saffron is very expensive and its adulteration is at high level, so there is a need of developing a system where we can detect adulteration on the spot. So in our future studies, we are developing a sensor-based data. We are using biosensors for on-spot detection of adulteration of saffron. So these are my references. Thank you.
Okay. So I would like to ask our scientific panel, do we have any questions for him? Okay, so we have one question from Dr. Neelkant sir. I think he says that like in saffron, you check which peaks are visible in graph and can ask which chromophore of saffron is giving the lambda max at particular wavelength.
According to ISO guidelines, there are three metabolites like crocin, picrocrocin, and saffronal. So crocin is responsible for color. So it is detected at 330 nanometer. Saffronal, it is detected at 440 nanometer, and this picrocrocin, it is detected at 257 nanometers. This is the protocol according to ISO International Standard Organization. We followed that protocol, and we were able to get these peaks using UV spectroscopy and this HPLC analysis.
Okay, thank you so much, sir. Our next presenter is Mr. Ankit Bishnoi, who is a student from National Forensic Sciences University, India, and he will be presenting on the topic Need of Drone Forensics, Getting India Ready for Future Drone Attacks.
Hello, am I audible?
Yes.
I would like to present my screen.
Yes, sir, go ahead.
Thank you. Is my screen visible?
Yes, sir.
Is my screen visible?
Yes,
okay.
Sir.
So, good evening to all the team members in the panel. I would like to take your permission to start my presentation. May I start?
Yes, sir. Please do it in full screen.
Okay, okay. Is it visible now?
Yes, sir.
Okay, so I will be talking about the drone forensics, why there's a need of drone forensics and why India basically need the drone forensics, because in the past we have seen in few days ago, India was hit by a drone attack wherein explosives were transferred from the border countries to the India. So aims and objectives of the studies will be, this study aims to ascertain exactly what data can be extracted from the UAV devices. UAV devices, I mean drones, like unnamed aerial vehicles. To compare the different models of the drones, like how one model of drone is different from other models in not perspective of the company and material, but also in perspective of storing that in digital information inside it. To study the usefulness of extracted data, like what data we can extract from it and how it can be useful to us. Also, we will be evading some detection anti-forensic techniques, like by which a attacker can try to absolute by attacker can absolute and how he can escape from us. Also, we will likely analyzing the storage media, where the storage of the drone material is basically goes in different drones. Introduction: the drones are referred as a UAV, unnamed aerial vehicle. These are the wide range of drones in the market today. We are equipped with different variety of drones. Drones are designed for different purposes. They are used by the law enforcement agencies, also for the disaster response, delivery, and for filming, like in the entertainment industry. But with the sophistications of the drones, attackers have started using them for their ill activities. Basically, the carrying capacities of drone are now increasing, and due to their specifications, people do use them for committing different crimes, like smuggling across the border, and even in the future we can see there will be drone mounted guns, and basically the smuggling across the border and in the prisons is seen very much in the past events. This study will describe the collection, examination, correction, and analysis of important artifacts recorded from a flight data. So in this slide we can see that how in the commercial drone takeoff in the US study they say that by 2025 how, from 2016 to 2025 we can see there's an immense increase in the use of the drones. Besides this there's other beside this we can see that drones, basically we can see there's a crimes in the different forms, and there's a world map which is describing all the drone incident that had took place. That and here we are talking about the different crimes that could be done by the drone: border smuggling, escape, eye prison, then immediating hackers, smuggling weapons, even the use of drone in restricted area. And not going away, India in first, it was the first India in drone attack in India. Drone used to drop explosive in Jammu Kashmir airbase. Now, how what methodology we could have adopted here? What I have adopted to done this study is like I use the data set of Video Labs. Video Labs is basically like a US organization which work on the different drones data. And beside this too, I acquired their images. I acquired the images because how a drone works is it is connected to a controller, and controller is basically the mobile device. So all the data from mobile controller and the drone is taken out. Is imagine, and then different tools that I used like datcon, csv view, exif, exd, kml viewer, google earth, drop, drone phaser, etc. Drop, drone phaser is very basically a tool used for like decrypting the dat file that do stored in the drone. Then as per my study, I have divided these artifacts in different parts, like user created data. They will be digital image and the video, audio files, and the waypoints of the flight. They are decided by the user. Then there will be user activity like the flight logs, the waypoints, and the motor speed, the height reached during the flight, and the flight path, and the launching and landing location. There will be device operated data, that will be FAA registration number. After 2016 there is a law made by the US government that all the like drones which are flying in the air should be registered. In India we are having such policies that we should be having our drone registered. Associated devices, firmware version, and all these information will be having. So I have divided it accordingly. So what all different drones are used, like I have used 5 models of DJI, then UNISIC, and then the Parrot. So basically what happens in the drone forensic analysis: we receive the request, we register the case, we register the exhibit, photograph the exhibit, conduct analysis.
There are 3 minutes left.
Okay. Then we have 4 method of extraction: physical, file dump, logical. I won't be going on that. I will be going further the critical and the data sources. We can have the SD card from the flight control, from the GPS and the sensor and the controllers. But we can have network information, the dat files, the text files, and all this with the we can have the sensor logs. Now coming to the tracing with the drone serial number. So basically, first of all, as you can see, there is every drone do contain the serial number at the outside it. As we technique, so anyone can absolute this anyone can erase this information. But what about the information present inside the drone? When you will be having, when you will acquire an image of the drone, you will see that drone. You can go you can use different tools, and you can use the normal visualization by which you can go to the device informations, as I have mentioned the path where we can get the different information. Here we can get the account V card, for example. It's synced with your mobile, so you will be having all the contact details there, device information, host name, serial number, and with that we can trace it. As you can see here, it's clearly shown that Android device was used, and it was Galaxy G7 Prax. Now coming to flight data, different drones have their data location at different. Some do use internal SD card, some use external use, and some are used like the cloud storage also. But in my studies I haven't been across drone that was using cloud storage. There's a different type of file type, .csv, then the default name like flight, and then with the using that cone, basically we can decrypt that file so that we can go the text file, and we can go the csv file. With the csv file we can use other software to convert it further to get about the drone activity. That what will happen, you can see that how the distance was covered and how it was reaching. Then with the KML viewer and Google Earth, we can upload that file and we can have a 3D graphical representation that how drone was going. As you can see, in the first we can see the timestamp, second we can see the altitude and speed of the during, third is the distance of the launching point, fourth is the download the date, then we can download the same file in different versions. Then the satellite view of the flight. Then running camel with Google Earth, same. We can also get, you can see that landing gear loading, landing gear raising. So how we can also come to know which was the landing and landing and launching point. So with all this, if we could have we can seize a drone, we can narrow down to 90% that who from where actually it was launched, who is the owner, and for what purpose it was used. So basically this is much needed in the border areas, and there should be strict law that that do cover all this study. So basically, to conclude, I will just say in my project I have conducted a comprehensive comparison drone forensic study with different UV
Sir,
and
time is up.
yeah,
I'm
just one
sorry to
second.
interrupt.
Okay.
Okay, so we will take questions.
Yeah, sure.
One is from Vikas Risdan. Sir, like what information we can obtain from Wi-Fi adapters used in drones?
Using the Wi-Fi adapter, we can download the PCAP files if there is a present in the if it is present in the drone I am talking about. Or this study basically says that all information is not present in all the drones. If there is a PCAP file, then we can further investigate like from which was the IP address used, from where the signals were coming, and further we can investigate the things as per the scenario.
Okay, the next question is how IPDR and CDR help us in drone investigation?
Basically, I like CDIR and IPDR, they have formulated rules which do we use for the forensic analysis. If you are talking about CDIR like the organization, or you are talking about something else.
Hello, good evening. Actually, I'm talking of the drone which is using the eSIM. If they are using the eSIMs, and if the signals are there from the suppose if we talk of the recent air drone attack in Jammu, and if they have been using this eSIMs, and the eSIMs are using the signals of the our neighboring country, then how it's not possible for us to get the IPDR and CDR details from the those towers which are across the borders?
So that scenario comes when international network will be having. For example, you're talking about eSIMs. So when we talk about eSIM, we will be using like different tools. That will be, and that part will moreover go to mobile forensics and network forensics. So for examining the
But in that case, the IPDR and CDR won't be available now. We have to approach it's the international outside the country. The towers are there and signals are, and they won't be giving us the IPDR and CDR details that can help us in the investigation.
Yeah, there is a law that every international laws that, for example, even if we having normal like cases in the country where people do use the eSIM to commit the crime. So I have been working.
Sorry, Vikas sir and Ankit sir.
Working with the law enforcement agencies, and it available because end of the day there is a virtual device. Even though it's a virtual device, it is somehow connected to network, and that network is coming from the service providers. So all these logs are available. It's just like we need to have law enforcement agencies permissions so that we can further investigate them. Hello.
Okay, now we will move on to our next presenter, who is Ms. Vartika Khare, research scholar from Bundelkhand University, UP, India. She will be presenting on the topic, a review on the advancements in chemical examination of composition of latent fingerprint residues.
Am I audible?
Yes, ma'am, you are audible.
Good evening, everyone. My name is Vartika Khare. I am research scholar in Bundelkhand University, and I am going to present a review on the advancements in chemical examination of residue of latent fingerprints, in which I'm going to cover the following topics. So coming to my introduction, fingerprint, as we all know, are the result of pattern left behind on surface because of sweat secretion by papillary skin ridges. The chemical composition of fingerprint is different in their composition because of origin from different glands of the body like pectorine, apocrine, and sebaceous. The detailed analysis of fingerprint in providing information about a person's age, gender, ethnicity, lifestyle, and occupation is important aspect of fingerprint examination. So the article aims to furnish updated review about the analytical techniques applied till date in the qualitative and quantitative examination of fingerprints and various influence factors which affect the constituents of fingerprint. The factors which influence the chemical composition of fingerprint, which we can see, is lifestyle, occupation, environmental condition, types of substrates, and external contaminants which we regularly get from the environment, and age of a person. So coming to lifestyle and occupation, in a study conducted by Dark and Wilson, they found that the presence of palmitic, stearic, and palmitoleic acid was found as the most abundant fatty acid, and also the presence of cosmetic from a female subject's fingerprint were found using GCMS technique. And occupation also play a vital role, as in a study, it was found that office workers were found to have highest amount of chloride in their sweat, followed by the laboratory workers, and finally workshop employees. Medications which we take in our daily routine also affect the constituent of fingerprint, as Jacob found in a study that methadone and EDTP, which is its metabolite, was found in the opioid-dependent patients by UPLC mass spectrometry. Sinkoska, in his study, found that detection of explosives like arsenic, nickel, and gunshot residue, and also the presence of drugs using the TOF SIMS technique on different types of surfaces. The drugs handled or taken internally could be distinguished easily using the constituents of sweat, and diseases also affect the constituents of sweat, as it is found that acne affect the squalene and fatty level of the sweat as found by Lamtowski. So variation also occurred with the age of a person, as we found in the study by Yamamoto that newborns are reported to have higher cholesterol and steroids level, and there is an increase in the levels of sebum during adolescence, which starts decreasing after adulthood because of reduced hormonal stimulation, as found in study by Pochi and Jacobson. The types of substrates also play a vital role in the identification and examination of fingerprint because it leads to the application of different chemical and spectroscopic technique for the identification and visualization. Alexandri tried to extract different amount of DNA from various substrates and found good results. Burley, in his study, studied acridine and sebaceous print on various types of surfaces using micro XRF and found good results. Environmental condition is a major influencing factor in determining the constituents of fingerprint because it was found that children's fingerprint in Oak Ridge National Laboratory, a study was conducted and it was found that children's fingerprint disappear faster from non-porous surfaces than that of adult. And in study by Bloodstain, the reason was found behind it that children's fingerprint contain large amount of volatile esters. In another study by ORNL, it was found that the presence of nicotine could also be detected from the constituents of fingerprint because the Buchanan in his study reported that the nicotine was found in the sweat of that person, but he had stopped consuming tobacco some time ago. It was found because he had handled tobacco products some time ago. In a study conducted by Ricky, it was
3
found
minutes
that
left, ma'am.
3 minutes are remaining.
Okay.
high temperature also affect the high temperature also affect the constituents of fingerprint. So as we have seen that external contaminants like cosmetics, hair products, or can be detected easily using the constituents of fingerprint. The instrumental technique which are widely used in the detection of constituents of fingerprint are usually GCMS, as Michalski and CAD classify different races and gender by using GCMS technique. The second technique used is FTIR, which was mainly used to image the fingerprints on different surfaces, and also was used by the scientists to determine the presence of various drugs and explosives. Mildly MSI technique was majorly used to study the degradation of constituents of fingerprint and detection of various compounds of drugs as well as cosmetics as found by Lawson in hand lotions. The top SIMS technique was widely used by Hinder and Simcosta as reported earlier, and they also detected external contaminants on various surfaces. Raman spectroscopy was mainly employed for the detection of explosive-related constituents, and immunogenic nucleic acid reagents are used widely nowadays by scientists as it leads to better results, and it was used by Russell to study the metabolites of cocaine to detect the constituents in a smoker's fingerprint. And similarly, cocaine binding aptamer were used by scientists to detect cocaine in the fingerprint. These are the combination of different types of techniques which were employed by the scientists in various years to extract the detailed information about the contaminants, contaminants in the fingerprint, like drugs, explosive, cosmetic products, etc. So I would like to conclude that advancement in the identification of chemical constituent of fingerprint is very vital, as it could reveal information about the person's lifestyle, occupation, gender, and ethnicity. Apart from the physical pattern, which is usually obtained from the crime scene, research is very necessary in this field, as developing of individuals' chemical profile could also add to the already existing information apart from the pattern. These are my references. Thank you.
Thank you so much, ma'am. May I request the scientific panel to ask questions? Okay, no questions. Thank you so much, ma'am. Moving on to the next presenter. Our next presenter is Ms. Saloni Malik, who is a student from National Forensic Sciences University, India. She will be presenting on the topic, the new player in diagnostics, Aptamers.
Hello.
Yes,
Am I audible and the screen is visible?
Yes, ma'am. Ma'am, can you do it full screen?
Yeah, sure. So good evening, one and all. Myself, Saloni Malik. So my topic for today is Aptamers, the new player in diagnostics. So these are the topics I will talk on. Aptamers. The name itself defines aptus means to fit and mers are the smallest unit of repeating structures. These aptamers are synthetic single-stranded DNA or RNA sequences elected from combinational nucleotide libraries through the well-known in vitro selection, that is selects. They have high affinity and specificity in identifying the target molecule. These have similar affinities as antibodies to the target molecules, so they are also known as artificial antibodies. Now let us compare these two. Major points are that aptamers can bind to any target molecule or cell, while antibodies are limited to immune responses producing targets. And the aptamers are small in size, while antibodies are larger in size. Aptamers have high specificity, functionality, and stability comparatively to the antibodies. So now moving on to the production of these aptamers. So how these are basically being produced. Uh, Generally, select cycle is being carried out. So in this cycle, there are three steps. First one is library generation. Here, artificial library is designed and oligonucleotides are being produced where are being there, so, which are around thousand in numbers, and then the second step is the selection where the aptamers will bind to their targets from the library, and other non-specific sequence will stay in the binding buffer, and then the aptamers are separated from the aptamer target complex by treating with the elution buffer. In this step, counter selection is usually implemented to eliminate those that do not bind to the target of the interest. Then the third step is amplification. The sequences eluted from the target are amplified by the employing PCR for DNA and reverse transcriptase PCR for the RNA to produce a sub-library for the next round of the selection. So these three steps consist of one cycle of selects. When the affinities of the sequences bound to the target are saturated, the sequence from the last round are sent to the clone and the sequence, followed by the identification of the aptamer sequence that bind the target with high sensitivity and specificity. So this is how aptamers are being produced. Now let us see the target binding mechanism of aptamers. Here the aptamers fold their flexible single-stranded chain into 3D structures which may change upon binding to their cognate target molecules. This structure switching characteristics has been capitalized in many aptamer applications. So these are the general application of aptamers, which shows it is useful for various purposes. So let us see the ways that aptamers can be helpful in forensics. First, by the use of the apta sensors. The most widely used apta sensor type is fluorescence-based optical apta sensor. Aptamers have been used as a bioprobe in optical sensor based on the incorporation of the fluorophore and or a nanoparticle. In the case of the fluorescence detection, the simplest format is to label the aptamers with both a quencher and a fluorophore. Aptamers beacons are probes that can monitor the presence of the target in solution using fluorophore quencher pairs. They can change in two or more conformations, such as hairpin shape or any hybridization form by the target binding.
Please conclude. 3 minutes.
So in this picture, we can observe that in the A figure, the binding of the target stabilizes stem and brings the quencher and the fluorophore in the close proximity. So fluorescence is decreased. While in the figure B, yes?
Ma'am, sorry to interrupt, but you have only 3 more minutes left. So
Yeah.
please conclude.
Yeah. The binding of the target brings the oligomers together and leads to the ternary complex stabilization. While in the figure C, it is being shown that the target binding induces an antisense displacement and results in the fluorescence increase. Another application is in the bioimaging. It is being used in MRIs also. And one more application is in the microbial pathogens from the crime scene. By this method, we can also identify them. And now moving on to the advantages and disadvantages. So we have seen that the aptamers are easier and more economical to produce with better binding ability, stability, modification. specificity and improved tissue, its susceptibility and to the renal, and few of its drawbacks are that it has the short half-life and the susceptibility of the renal filtration. It has the susceptibility of the renal filtration. And finally, in the conclusion, I would just say that these aptamers may be the significant benefits to the forensic analysis, allowing the rapid, sensitive, and specific detection of molecular targets relevant to the criminal investigation, forensic drug analysis, and the forensic toxicology. These are my references. Thank you.
Thank you so much, ma'am. Any questions from the scientific panel?
I have a question, please. Aptamers are very interesting in the forensic application, especially for the biological detection. But do we have any data if they can make some potential bad effect when we, after the identification, we run the DNA analysis? Is there any literature or data about this point?
So far, the papers which I have referred, and as per my knowledge, I haven't found any of the drawbacks or the wrong effects on the body.
Okay, thank you.
Thank you, sir. Thank you, ma'am. The next question comes from Rajveer, sir, who is the Deputy Director of Rajasthan FSL. He says aptamer can be used in treatment like cancer cells.
Yes, it can be used for the treatment of cancer cells, and in fact, in many diseases, it can be used.
Okay. Next question is from Dr. Hardy, ma'am. She asks, which are natural aptamers and what are some examples of riboswitch?
I'm very sorry, I am not aware about this.
No problem. The next question is from Dr. Neelkant, sir. Stability of aptamers is a big challenge. How will you address this?
Maybe because it can withstand in the normal temperatures and can act in the normal way as of the antibodies, and which helps even in the target delivery of the proteins.
Okay, ma'am. Thank you so much. I would like to tell my audience that we are left only with 2 oral presentations. So moving on, I would like to call Ms. Jasra Rahman, a student from LNJN National Institute of Criminology and Forensic Science, India. She will be presenting on the topic Effects of Medium of Primary Education on the Learning and Usage of Latent Script.
Good evening. Am I audible?
Good evening, ma'am. Yes, ma'am.
Is my PPT visible?
Yes, ma'am.
Okay. Good evening, everyone. I'm Jasrana Sadiq Rahman. My paper topic is on the effects of medium of primary education on the learning and usage of Latin script. We know that the Indian education system, just a second, I think my PPT is stuck. Okay, yeah. We know that the Indian education system caters to its diverse population, where only about 10% of the primary schools use English as a medium of instruction. Not only the 22 major languages recognized in the Indian Constitution is taught in the primary level, but also the needs of the linguistic minorities are met. As the Latin script followed by English is much different from these languages, there is a difference in the writing style. This difference is referred to as the accent in handwriting. Like accent in speech, the accent in handwriting is influenced by the writer's previously learned writing language. In this case, the medium of instruction in primary schooling. From a forensic point of view, this detail can be used in cases where admitted samples present are only in a language other than that of the question documents. This study narrows down the suspect list in cases of unknown authors in anonymous, disguised, or forged documents based on the author's ethnicity or at least the region of primary education. Interscript comparison of handwriting has been studied before, where it is proven that writers carry some features from their vernacular languages into their English writing, which is seen as idiosyncrasies, as stated in the first paper that I have given here. This comparison is not only done using classical handwriting comparison methods, but also digital aids and statistical models. It has also been discussed that the analysis by nature opposes the principle of comparison, that is, like is to be compared with like. Thus, it should be kept in mind during the analysis. For this research, samples were collected through from primary source through questionnaires distributed at convenience through friends and family. The questionnaire composed of details like the name of the subject, language of the primary education, frequency of using the said language over English for writing, and the signature of the subject for authenticity, along with the handwriting samples. For the English sample, the exemplar National Park toll letter was asked to write, as it contains all the letters of the English alphabet and the numbers from 0 to 9. This provides an adequate picture of the writer's habits such that a meaningful comparison can be conducted. For the language of their primary education, a simple paragraph was given considering the ease of translation. In total, a sample size of 30 was collected in 8 languages of primary education. These are namely Assamese, Bengali, Bodo, Hindi, Manipuri, Marathi, Kannada, and Urdu. An overview of the scripts of these languages was done to understand its origin and interrelationship. It is noted that Assamese follows the Kamrupi script and is a sister language of Bengali, where all the letter formations are similar, except for 2 consonants that exist in the Assamese alphabet, but not in Bengali. Bengali follows the Brahmi script, which is the parent script of Devanagari, and Hindi and Bodo follows the Devanagari script. Marathi follows a modified version of Devanagari called the Balabod, while Meitei, that is recognized as Manipuri in the Indian constitution, has its own script, which is a descendant of Brahmi script. It has influences of Latin script as well. Kannada follows a Tamil Brahmi script evolved from the Dravidian family of languages, while Urdu has a Nastaliq script, which is a popular calligraphy style of Persian alphabet derived from Naskh script. These samples are analyzed using reflected and oblique lighting, and the writings in English are compared to the writings in the other language of the same author. Here we see the class of characteristics observation in a tabular form, where the number of authors having different class characteristics in the 2 writing is compared to the number of authors who have the similar class characteristics in both the writings. The class characteristics that are considered here are slant, alignment, speed, initial and terminal strokes, and line quality. We note that the greatest difference is seen in the speed and the angularity of the strokes. Other characteristics like connectivity of strokes is not considered, as there is no basis of similarity between the given languages. For individual characteristics, only letters with similar formation or embellishments are considered, so that it is in agreement with the principle of comparison. We see in the class characteristics that the greatest difference is in speed. This can be attributed to the frequency of writing one language over the other, while the difference in the angularity
3 minutes
can be
are left.
explained. Okay, yeah, thank you. While the difference in the angularity can be explained as the sample size of Assamese is major and Assamese script demands the angularity in the letter formation. All these characteristics and the strokes of letter size and proportion of the writing and the formation and position of embellishments shows that an author does not break free from the writing style, even across scripts. This proves that handwriting accent prevails for anyone who has learned the different script prior to learning to write in English. We have to note that this study does not take into consideration the writer's condition, writing condition, and any other factor that might affect the writing. Also, it cannot determine the first learned language or the language of primary education solely based on the English handwriting of the writer. But given the writings of an author in 2 languages, it can establish that both the writing are done by the same author. And considering the great number of schools imparting education in non-English languages in our country, our perpetrator belonging to one of the schools has a higher chance, thus narrowing down our suspect list and helping some progress in the cases of question documents. Thank you. These are my references.
Thank you so much, ma'am. Now I would like our respected scientific panel if they have any questions for you. Okay, so there are no questions. Moving on towards our next presenter, who is, so this is the last presentation of the session. Moving on towards our next presenter, Dr. Abhishek Kumar, who is a student from NFSU India, and he will be presenting on the topic crime mapping, constructing a safer community.
Am I audible?
Yes, yes, you're audible.
Okay. So, good evening, everybody. I'm Dr. Abhishek Kumar, MSc Forensic Odontology at the School of Medical Legal Studies. Today, I'm here to talk about the subject crime mapping, constructing a safer community, which I have done under the guidance of Dr. Dhani Patel ma'am, who is an assistant professor at the School of Medical Legal Studies, NFSU. So, my first slide is introduction. Basically, crime mapping is a tool used to identify and highlight suspicious events that may require further investigation. It actually locates the location where crime has happened, and it also displays the area in a graphical representation. It also displays the crime area in a graphical representation. The main objective is to identify the hotspots. Hotspots are basically high concentration areas of crime. We would be hearing about hotspots a lot whenever we talk about crime mapping. So coming to the history, in 1829, the first crime map was created by Adriano Balbi, who was an ethnographer, and Andre Michel Guerry, who was an attorney. And they basically created a crime maps on topics such as property, crime against people, and levels of education. In 1831 and 32, Lambert et al. Published 3 maps on the same topic, but it was spread across the larger areas. In 1920s and 30s, the first crime map in USA was created by a group of scholars from the University of Chicago, led by Robert Park. And in the late 1960s, the automated crime mapping came into the picture. And since 1980s and early 1990s, desktop crime mapping has become commonplace. It's become a very usual thing. Coming to the evolution of crime mapping, earlier pin maps were used, which consisted of pin maps, color dots, paper maps, push pins. It actually showed that where crimes have occurred, but it also had certain limitations. Next came the mainframe computers. Mainframe computers were basically, if they used whenever crimes were mapped on mainframe computers, it was a very labor-intensive process. Then came desktop computer mapping, which is a very usual thing now. Desktop computer mapping was basically started first on used by military, but now it has become a very common because it is used commonly by the police nowadays. Now, crime mapping also involves the use of computers and GIS. GIS is Geographical Information System, which is an organized collection of computer hardware, software, geographical data, and personnel designed to efficiently create, store, update, manipulate, analyze, and display all forms of geographically referenced materials. It also helps to identify the crime hotspot areas. It can also explore the link between the crime and the environment. Along with the crime mapping, GIS also prepares the police department for future crime problems. Coming to the importance, it displays the spatial crime events, and as I have already stated, that the main objective of crime mapping is to identify the hotspot areas. It also helps to integrate community characteristics so that it can formulate a relationship between the crime and the community and the characteristics of the community. And then it also helps for spatial analysis with selection and query tools. Next, coming to the future prospects. As this is a new subject, I see a lot of future scope in this. That prediction and emergence of future crime hotspots may happen, and also a new methods of map evaluation and adaptation of the practice in different policing cultures can also take place in future. Coming to the conclusion, crime mapping is a preventive measure to crime, and due to recent advancements, there is a spotlight on crime maps when it comes to prevention of crime. It also ensures the safety and security of the region and the people. It also facilitates the spatial decision making for the enforcement agencies for better surveillance, scheduling, and planning. These are my references, and thank you.
Thank you so much, sir. Do we have any questions from the scientific panel? Okay, so with this, we complete our oral presentations. I would like to thank everyone, like the dear participants, for presenting their valuable work with us. The results for this presentation will be shared shortly on our YouTube channel within 2 to 3 days. As rightly said, if you can speak, you can influence, and if you can influence, you can change lives. Well, it does not matter who wins, but it's important to keep participating. So I wholeheartedly congratulate all our participants. So let's keep trying and keep learning.
So with this now the time has come to reveal the winners of a quiz. Therefore, I request Mr. Robert Skal, sir, to please tell us about the 5th winner of the quiz.
The 5th winner is Preeti Sangwan from Maharshi Dayanat University, Rohtak, Haryana. And I apologize for any mispronunciation.
Congratulations, Preeti. I request Dr. Eugenio, sir, to please tell the 4th winner of a quiz.
Yes. The 4th winner of the quiz is Miss Shriyaze Chattopadhyay from Sri Lakshmi Narayan Institute of Medical Science. I apologize if my pronunciation was not totally good. Congratulations.
Congratulations, Shriyaze Chattopadhyay. Now I request Dr. Rajesh Babu, sir, to tell us about our I think he's...
Yeah, it was very nice. Really, I didn't expect, honestly to say, I really didn't expect that this much of an enthusiasm, a dedication, a sort of in-depth survey that the students would have done. Really, it was heartwarming for me. Of course, it is not in no way I am demeaning or anything. I felt so elated when I see all the students presenting so enthusiastically and so sincerely in in their respective topics. Of course, it was really, I should say, more and more informative. And rather than that, even in, I feel some of the students who are in even bachelors also, they are able to present in a very, very splendid manner. Really, it shows the, what I can say, a sort of more and more awareness and also enthusiasm in the updates of the current technology in forensic sciences and their dedication, I should say. It is very much, I was moved by the way the students have presented. I mean, every presenter, I mean, I'm not singling out any particular student. The entire presentation, entire group of presentations, whatever I had come across, it was really splendid. Thank you so much for having been in this particular platform. Though we are unable to do it in the physical, but still in the online also, we feel as if we are among us. So really, it was very nice to have organized such a conference in such a short period of time. Very
Thank you
nice.
so
Thank
much,
you.
sir. Thank you
Wishing
so much,
everyone
sir.
all the best. Okay.
Thank you so much, sir, for your blessings. I request Eugenio, sir, to please tell us the third winner also. The third winner of our quiz. Okay. So with this, I request Cloyd Steger, sir, to please announce the third and the second winner of our quiz.
Okay, I don't have the third winner information in front of me, but the second winner is Ashwara Chandran from Nurul Islam Center for Higher Education. I'm sorry, I don't have the third winner information.
Okay. So I request Professor Dr. Michael Freeman, sir, to tell us about the first winner of the quiz.
I think the sir has become kind of technical. So it's texted the winner to the team group. So could you please just check it and announce the winners of the first and third one as well?
Sir, Eugenio, sir, can you please tell the winners of third and the third one and the first one once again?
Yes, sure. I have in front of me actually. So the third winner, I will please correct me if I will make any mistake.
Yes, sir.
Is Miss Arvind T from Jain University. Is that correct?
Congratulations. Congratulations, Arvind. Okay.
And the first winner is Jewel Mari Sajo from Rashitra Rakash University, Gujarat.
Okay.
Congratulations.
Congratulations, Jewel Mary Saju from Raksha Shakti University. Thank you so much, sir.
We will soon be disclosing the winners of the oral presentations through our YouTube channel. So I request you all to subscribe to our YouTube channel to know the results. Also, we are in the process of launching more of such exciting events really soon. So for that reason, stay tuned with us. Thank you to our valuable audience for being so interactive and for telling us about your valuable comments. We will soon be revealing prizes for you all.
Thank you, Ashika and Nandini, for this wonderful anchoring. And I would also like to thank the scientific panel and all the audience that is present here. So now it's time that we are at the end of the concluding of the event. And as rightly said, a dream you dream alone is only a dream. A dream you dream together is a reality. Once again, on behalf of my whole team of the Forensic Corner, I'm extremely delighted
I think there is some network issue from Prashant's side. So I will conclude. So on behalf of my whole team of the Forensic Corner.
So you can carry on.
I'm extremely grateful towards each and every one of you who have made this event possible and added grace to it, annexing from the never-ending support of our very honorable Vice Chancellor, Dr. J.M. Vyas sir, Campus Director, Dr. S.O. Zunare sir, and all the esteemed faculty members of National Forensic Sciences University, Dr. Rakhi Agarwal ma'am, Dr. Bhargav Patel sir, Dr. Rajesh Babu sir, for giving their valuable time for addressing each and every one of us. Moreover, I would like to express my heartfelt gratitude to all the international experts, Dr. Emilio Nozolice sir, Robert Scal sir, Professor Michael Freeman sir, Dr. Eugenio sir, and Cloyd sir for giving their valuable time from their busy schedules for this conference. We will be looking forward to learning from you under your guidance. Furthermore, I would like to congratulate each and every single participant who come forward and participated in our conference. Without you, this event would be incomplete. Last but not the least, I would express my gratitude towards such an enthusiastic and patient audience. Without such an amazing audience, the event would not have been a huge success without your patience. So thank you so much, everyone, for believing in us and supporting us throughout.
Also, I would take this opportunity to further congratulate my team, beginning from Nandini, who depicted the real example of hard work and being the constant pillar for the event success. Without her without her presence, the event would be incomplete. Then finally, I would like to thank Shankar, our technical head, whose enthusiasm always surprises me. I learned a lot from him. His extensive technical knowledge is the biggest power and still surprises me. Without his technical knowledge, I think the event would have not been the way it is, what we see right now. Then thank you, Manisha, for your efforts and time for giving a virtual representation of the event. Your skills of designing made this event even more powerful and even more beautiful. Next, I would like to thank our wonderful anchor, Ashika, for being the voice of the event. You gave us the most outstanding heartbeat of the event. Thank you for trusting and supporting the event. Then I would like to thank Vidushi and Apurva for their support whenever we needed their assistance. Then finally, I would like to express my gratitude towards Shamik Bhatt for adding an asset to the event with his hard work and dedication by getting the blessings of Honorable Chief Minister Rupani Sir of Gujarat State, India. The success of the event is all because of our team effort. Without this wonderful team, we could not have done anything. Also, special thanks to Naman and Sanpreet, who gave identity to the students' initiative, the Forensic Corner. You are the real kingmaker and our back-end support. Also, I would like to give a big round of thanks to Indian Forensic Students Council for helping us promote this event, and I wish you all the success with your aim to promote education in the field of forensic science from your initiative. I would also like to thank Anika Aze, founder of Pink Biology YouTube channel, for being such a great media partner. Again, from bottom of my heart, I thank you team, the international experts and the National Forensic Sciences University national experts for trusting and supporting me for this event. It's all because of your collaborative efforts we could make this event a huge success. Also, I would now request our international and national experts if they would like to share their views and their comments regarding the event. And in case if I did any mistake or my team, I wholeheartedly apologize to the audience and to our scientific panel.
There was no mistake at all, Prashant. Everything was very flawless. It was flawless. I'm very proud of you people. The entire team deserves much accolades. Okay, keep it up. All the best in future too. Okay.
Thank you.
Thank you, sir.
I would like to thank you for allowing me to participate in this, and I congratulate you. I was very impressed with the success of your event, the enthusiasm of the participants. You're doing good work, and it's a pleasure to work with you.
Same from my side. I want to extend my congratulations to Forensic Corner and the university and all the colleagues and students that join the conference. It has been a very important and precious time for me, hopefully to continue on this way. Good luck, guys.
Thank you so much, sir. Also, I would like to thank the Deputy Director of Rajasthan FSL, Rajasthan Forensic Sciences Laboratory, Rajiv sir, for being the constant valuable participant in this event. And sir is still giving his blessings to us through the chat. If sir could also share some views, it will be really nice, sir. And thank you so much for constantly being at the event.
Hello.
Yes, sir.
You getting my voice?
We are able to hear.
It was a nice conference, and it's a good effort to, you are involving international expert from all over the world and the national expert for this conference. Definitely, it is a big success of this event. For the future, I'm giving best wishes. As far as the participants, the level of participants is concerned, they are very much, it was a high level participation. So all the best for the future.
Thank you so much.
Thank you so much, sir. And before we now, the most awaited moment, before we release the surprise to our audience, I would also like to give my thanks, a vote of thanks to our national faculty members, National Forensic Sciences faculty members, Dr. Hardy ma'am and Aditya sir, and also Dr. Neelkant sir, for constantly being present at the event. And in case if I'm missing any expert out and any of our valuable participants, I'm so sorry because it's a huge crowd. And also lovely audience, thank you so much. And now Nandini, it's time that we can release the surprise which people are waiting for, and especially our commenters, because they are waiting at YouTube to find out that what is the surprise actually. So let's reveal.
Yeah. So, okay, as promised, here I present a very big surprise for all of us. We would like to thank the editor-in-chief of the Forensic News Journal magazine, an international magazine of Philadelphia, USA, Sonia Trippett ma'am, who will be releasing a special issue of the first ever International Forensic Science e-conference powered by the Forensic Corner and organized by the students of National Forensic Sciences University. This is special issue in her magazine showcase the winners of the quiz along with their articles and all our event proceedings, including the list of our esteemed national and international speakers. It will also be showcasing the winners of the oral presentation. Along with that, it will also contain the names of the persons who asked the best questions from our audience, with the speakers and much more exciting things. So please stay connected with us. Once the magazine of Sonia ma'am is released, we will be circulating the links with all of you. And once again, thank you so much, everyone. We will see you soon with another Power Packed event. Thank you so much. Now I request our
Thank you so much, respected
with us.
Thank you so much.
Shankar, it is requested that we can end the live stream so that the international and national panels can discuss regarding the further proceedings of the event.
Sure.